I'm really happy to be here today. I wasn't supposed to start, but I think we're at a very, very critical time for science and for patients. And I really want to welcome you and tell you about how important about the work you're doing and what you are doing for patients. I want to also thank our friends at Parker. We're doing this in collaboration with them, and they have been great partners, and thank you very much. This meeting comes at a moment of extraordinary scientific promises. Therapies are offering real hope where none existed before. But these therapies are not just breakthrough in science, they are lifelines for patients and hard-to-treat cancers. And I don't think I have to remind anyone in the room that we are all working to help patients, and that is our obligation and our responsibility to get the best science in these diseases that are still haunting them. But this promise won't fulfill itself without requires all of us working together to rethink and how we develop manufacturing and deliver these to patients. At a time when critical institutions are under strain, your presence today sends a clear message. We are committed to ensuring science continues to move forward and that it reaches the people who need it the most. I think everyone in this room knows that, and we have to be advocates and speak up and make sure that the right science, when appropriate and is safe for patients, gets to them. I want to thank all those at Forensic Cancer Research and Jeff Allen and everyone that has worked together on this, but I welcome this conversation and we hope for some outcomes. So thank you very much, and I so appreciate what everybody is doing.
Good morning. Thank you, Ellen, and let me add my thanks for everyone for joining today's meeting. We are very pleased to have you here, and on behalf of our co-host, the Parker Institute for Cancer Immunotherapy, we're very proud to offer today's meeting and look forward for the discussion to come. This important discussion today will focus on a new set of proposals in the rapidly changing cell therapy space by reimagining clinical trials, manufacturing strategies, and treatments for patients, particularly in the context of rare diseases. Today's not the first time that we've had the opportunity to partner with the Parker Institute. In fact, this has become a bit of a series over the last couple of years. You may recall that last year's meeting was a focus on strategies to identify and testing gene modification candidates in early clinical trials. The manuscript from that forum is well on its way, and we expect it to be published very soon, so please keep an eye out for that. Identifying promising gene candidates is a step toward effective therapy, but that's just the beginning. So to extend on the work from last year, earlier this year, Friends and Pisces pulled together a multi-stakeholder working group to develop a practical framework for advancing regulatory and manufacturing approaches that can enable timely access to promising therapies while ensuring quality and safety. This includes the potential for regulatory flexibilities to facilitate cell therapy development in small populations, cost recovery considerations to engage payers earlier in the process, and scalable manufacturing approaches, including the role that aspects of decentralized models might be able to play. Today's conversations will examine operational, safety, and quality control strategies to support flexible, risk-adjusted manufacturing approaches while maintaining rigorous standards. I want to thank the whole team at the Parker Institute for their partnership in this effort. And in addition to the panelists today, numerous experts have been involved in the working groups leading to today's discussions, and we hope that you'll take the time to review the white paper that was distributed in advance, capturing their thoughts and ideas that will be the basis for today's discussion. We thank all of the participants for their time and expertise that have led up to today's meeting. Following each presentation and the panels for today, we'll have the opportunity for audience questions. We ask you to please utilize the Q&A link and a QR code that should be available on the agendas at your seat. This should be relatively straightforward. All you have to do is scan the code, enter your name, and you'll be able to enter your questions electronically. For those of you that are joining virtually, please utilize the chat function in order to submit your questions, and members of the Friends and Parker teams will be monitoring to work and get as many questions answered and to the moderators as we can. Now, to kick things off for today's discussion, I'd like to turn things over to Dr. Crystal Mackel, who will be giving an introduction presentation ahead of our first session discussion entitled Advancing Regulatory Frameworks for Cell-Based Therapies. Thanks, Crystal.
Thank you. Good morning, everyone. Thank you for taking the time out of your busy schedules to be here. I want to in particular thank Ellen Siegel, Jeff Allen, Mark Stewart for your expert leadership, and all of the rest of the staff at Friends for shepherding this effort thus far, as well as the great people at the Parker Institute for Cancer Immunotherapy who are dedicated to bringing cancer immunotherapy to all patients who can benefit. So let's see. Oh, my slides. Thank you. Relying on Mark to get them there. All right. Great. So cell therapies for cancer, there's no question that they have had immense impact. And I think there are all signs are demonstrating that they're poised for more impact. The question is, how much more? And it is our belief that we're just getting started. It took 40 years for tumor-infiltrating lymphocytes to move from a theoretical concept, first at the National Cancer Institute, finally to FDA approval last year. Beginning in around 2010, we saw chimeric antigen receptor-based therapies mediate dramatic and sustained cancer control in patients with B-cell malignancies refractory to all standard therapies. This led to pharma investing in cell therapies, which was really a heretical idea not so long ago, and eventually creating commercializable products that are cell-based therapies. We've now seen numerous CAR T cells approved. We've got 12 approvals across 5 disease indications, and now 6 different companies. And we're beginning to see proof of concept that this can extend beyond B-cell and plasma cell malignancies and into solid cancers. Now, all of this success thus far is remarkable, since this really represents still what we would call version 1 engineering. And in the laboratories across the world, we're seeing remarkable advances in synthetic biology and cellular engineering that are providing countless opportunities to make these products even more potent. So I think it is very reasonable to believe that we're just getting started. Now, what about children's cancers, where I've spent most of my career? I think we all know and have been inspired by the remarkable story of Emily Whitehead. She was the first child to receive CAR-19 therapy at the University of Pennsylvania in 2012. She received this therapy for B-cell acute lymphoblastic leukemia, a disease that is described as eminently curable, except for those patients for whom standard therapies don't work. And Emily was one of those patients. There were no options remaining for the treatment of her disease. She received 1 infusion of CD19 CAR, and you see her there, around the time that she received the therapy, and she remains in complete disease remission, shall we call it cure, to this day. And we see now how Emily has thrived after receiving this curative therapy. And she is now, a healthy young woman, I think matriculating at the University of Pennsylvania. And I think we can all say, wow, what a miracle. We need more miracles like Emily Whitehead. Well, one can argue that, progress, as I mentioned, has been really quite impressive. We now have these 16 different approvals for cell therapies for cancer. You note that, again, they represent version 1 engineering. They're relatively restricted to CD19 or BCMA. Only recently we now have the TILs and now a MAGE-A4 restricted TCR. The other thing to note is if you look at the age of the patients who are eligible to receive these therapies, you see that all but 1 require greater than 18 years. These are therapies designed for diseases that occur in adults. Indeed, what we saw with Emily led to the approval of Kymriah. This was the first cell therapy approved for the treatment of cancer, and believe you me, we certainly all were popping our champagne in 2017 because we thought, wow, this is the beginning of something very big for children's cancers. Unfortunately, it hasn't moved forward in terms of any other therapies. Now, it's not because the science hasn't delivered. There are several CAR therapies that are showing remarkable progress in diseases that afflict children. CD30 CARs for Hodgkin's disease, usually a curable disease, but for those patients who relapse, there are very limited opportunities, and you see complete response rates there of more than 50%. What about neuroblastoma? This is the most common extracranial solid tumor of childhood, and for patients with high-risk disease, less than 50% of the children are cured with standard therapies. Malcolm Brenner at Baylor, back in 2008, before Emily was treated, delivered a first-generation CAR T-cell targeting GD2 to patients with neuroblastoma, and several of those patients had impressive responses. They recently reported a follow-up on that, and you see 18 years of follow-up, and we still have patients who are cured from the GD2 CAR therapy. This is arguably the first evidence of cure of solid tumors with CAR T-cell therapies. More recently, an investigator in Rome treated a series of children who are refractory to all standard therapy and had a very sizable incidence of durable CRs you see there on the right. What about T-cell acute lymphoblastic leukemia? This is the other kind of leukemia for which there really are no targeted therapies. Here is a PET scan showing a disease completely infiltrating the liver and spleen of an adolescent, completely cured, or at least complete remission with CD7 CAR T cells. This is the disease that my group has been working on, diffuse midline glioma. This is the most common killer of children due to brain tumors. Despite decades of clinical trials, we have no standard therapy for this disease beyond palliative radiotherapy. When a child is diagnosed with this disease in 2025, the pediatric oncologist walks into the room and says, "I'm so sorry, please, we'll give the radiotherapy, but you really should take your child home and have some quality time." The median survival is 9 to 11 months, and no children survive beyond 5 years. We've been able to show using a CAR targeting GD2, we've got significant rates of disease regression and one durable CR about 4 years out. This is a GPC3 CAR targeting pediatric solid cancers coming out of Baylor. It didn't work so well until they put IL-15 in it, but you can see the armored version of it actually is inducing pretty sizable rates of response. And finally, the CD22 CAR, which has a very high rate of disease remission in patients refractory to CAR-19. So why aren't these being developed in pediatrics? Well, the issues, as we know, relate in large part to the incredibly high costs of developing and delivering cell therapies. If you do a little Google search and say, how much does it cost to develop a cell therapy today, you get a number of between 500 to 600 million. I venture to guess, Kristen might tell me that's an underestimate. We know it's a lot of money. And even for common diseases, these high costs of development are limiting investment. While we saw a very frothy time for investment around 2020 and 21, what we're seeing now is diminished investment in the entire sector of cell and gene therapy, and this is hitting the development of CAR T cells very hard. The problem of high costs of development are limiting innovation across the spectrum, and this problem is magnified for rare disease. Let me take a case study here. This is not cancer, but this is a rare immunodeficiency that results from mutation in the gene that encodes the gene Artemis. These severe combined immunodeficiencies afflict babies, and they develop multiple infections, and unless they are treated with something, will never survive infancy. While immunodeficiency is a rare disease, once you divide it into the individual genetic subtypes, they become ultra-rare diseases. And for this disease, it afflicts about 3 to 4 patients per year. The standard treatment is a hematopoietic stem cell treatment, and we all know the toxicities that carries, lifelong toxicities. And so investigators at UCSF, and this is really the work of Mort Cowan, Jennifer Puck, and Brian Shai at UCSF, developed a lentiviral gene therapy. So you take the stem cells from the bone marrow, you transduce them with a gene to encode the Artemis gene, and reinfuse them to the patients after low-dose chemotherapy. As of 2025, 17 patients have been treated, and 100% of them have demonstrated essentially complete correction of their immunodeficiency. They have transient drop in their blood counts, but no mortality and no chronic effects related to treatment. And so this is essentially a cure for a life-threatening immune disorder. Now, in order to be able to sustain this globally for all patients who need it, there needs to be a way to get this drug approved and to get reimbursement. There's no way that this can be delivered globally to all the children who need it using a perpetual IND, because research budgets are not designed for such a thing. So UCSF sought to, let's see whether we can file the BLA and get this thing approved. They have enough vector in their freezer to treat about 50 patients. Given that there are about 5 patients per year in the US, this would last them about 10 years. They know that this vector is stable for 6 years. Now, when they approached the FDA to talk about a BLA, so-called Type C meeting, what they learned is that in order to treat on a pivotal trial and to ultimately then, post-approval, conduct commercial manufacturing, they couldn't use the vector that they originally had. They needed a vector that was manufactured according to different standards. This was a GMP vector, but they needed a vector produced according to commercial standards, and they needed to produce additional lots of that vector to prove that they could make the multiple lots, the so-called PPQ runs. So when you add it all up, if they were able to use the GMP vector, which had cured their patients, it would have cost them, somewhere between $1 and $4 million to manufacture the vector. And by the way, they already had it available. But in order to meet these standard requirements for commercial manufacturing, they were looking at somewhere between 16 and 57 million just in upfront costs to create the starting material. This is an example of some of the challenges that we face in the field of rare disease for cell and gene therapy, where the standards are devised for common diseases, where you can imagine multiple batches being made per year. It is their belief that they could produce a single batch of vector that would last them for 10 years. And so I think what we're hoping is that we can begin to think about approaches that would proactively address potential sources of variability, which is what these commercial standards are all about, but do it in a way that is risk adjusted for rare disease. So there is an increasing focus on enhancing therapeutic development for rare diseases. Why is this? Because if you add all the rare diseases up, they become not so rare. Cancers and other rare diseases are among the top 10 causes of death for children in the US every year. An estimated about 7,000 children die due to rare disease in our country every year. In addition, as we've learned more about the molecular biology of disease, let's take cancer for instance, what we know is that we do best when we develop targeted therapies. But if you start to slice and dice lung cancer or another cancer into subsets, and you have molecularly targeted therapies, you end up with smaller subsets of patients to treat. And this is amply illustrated by the evolution in T-cell receptor-based therapeutics, an area of rapid growth and great opportunity. But TCR-based therapies are relevant only for patients with a particular allele. And in some cases, it could be an N-of-1 therapy for patients who have a particular neoantigen. Now, the FDA has clearly leaned in to this problem of rare disease, and they have been, I think, great thought partners so far. The Rare Disease Innovation Hub at the FDA was launched in January of 2025 with goal number 1 is to advance regulatory science for rare disease. And just in the last few weeks, we saw the very exciting news from Commissioner Macquarie, who is proposing a new path for drug approval for ultra-rare diseases. And we look forward to working with the FDA to enhance this progress in a responsible manner that increases the chance of cure while maintaining the highest standards for safety. Thank you so much. Oh, wait, here we go. So our focus and goals today. We're going to discuss regulatory manufacturing and cost recovery strategies to address the barriers that prevent promising cell therapies from reaching patients with a focus on cancer. Our goal was to inform policy discussions, highlight areas for regulatory clarity, and identify operational solutions to support sustained therapeutic development. Key focus areas: regulatory engagement and flexibility, manufacturing adaptability, and sustainable pre-market access. And so I want to welcome the first panel up. It's going to be led by Michael Kalos, and panelists are Lee Fleischer, Kristen Heggie, Holly Fernandez-Lynch, Nicole Verdon, and myself. Oh, there you go, Nicole.
I think you're down there.
Thank you.
I think, am I here?
I think you're next to Lee. They want us by.
Am I here?
By order. Well, good morning, everybody. It's a pleasure and an honor for me to moderate this first session on behalf of Friends of Cancer Research in PICE, where we're going to discuss the regulatory pathways to support access to cell-based therapies. I'm Michael Kalos. I'm Managing Director of Next Builder Consulting. And my panelists are Lee Fleischer, who's representing Rubrum Advising, Kristen Heggie, who's an independent board of directors member, Crystal Mackel, who you heard from Stanford University, Holly Fernandez-Lynch from the Perelman School of Medicine at the University of Pennsylvania, and I believe Nicole Verdun from USFDA. So this is going to be a 60-minute session that has 50 minutes of questions, 50 minutes of discussions, and 10 minutes of questions. Herding cats here. So starting out, we're going to start out talking about the current experience. The examples that Crystal presented highlighted the core tension that we're going to explore in this panel. How do we create regulatory and policy frameworks that enable access to therapies in high-need settings without, of course, compromising safety or opening the door to misuse? So to kick off our discussion, I think it's relevant to discuss what do Crystal's examples tell us about where the system is falling short today. She touched on that, of course, a little bit, but we're going to touch on a little bit more in this session, and where there may be opportunities to improve the system. So Crystal, if you'd like to kick off with your thoughts on that. Crystal.
Crystal.
Yes.
Oh, I think they've heard enough from me.
Yeah.
I think the science isn't holding us back. I mean, I've been doing this for 40 years. It's always been the science. Yeah. We need more scientific research, and that will deliver more cures. And I'm just dumbfounded to find myself in a situation where the science is going forward like gangbusters. We have therapies that can benefit and cure patients, but we can't get them to the patients. And how can we right-size the regulations so that we can create an economic model that will be sustainable for small markets?
Mm-hmm. Nicole, some thoughts on from your side?
Well, I think that the right-sizing regulations, it's a bit nuanced. I think that they're, from an FDA perspective, seeing a lot of programs and sort of really taking a step back and analyzing where are we falling short or where are there disconnects. I do think that there are some opportunities for earlier thought in terms of the CMC and manufacturing piece. And sort of thinking through some of the barriers and to sort of getting to the finish line that we see. One is you really need to have an understanding and characterization of your product such that when it does happen that you want to switch, your manufacturing from like let's say, for example, an academic setting to a commercial setting, that there is enough understanding such that you can get that done. We've seen a lot of programs get sort of hung up where they have to switch manufacturing because of, maybe a facility change or otherwise, and that sort of holds things back. I also think that we need to just have earlier conversations around some of the CMC pieces. We have a lot of flexibilities that we have at FDA in terms of programs and interactions to help with that. But I think that there's some communication that can be probably improved earlier on in the cycle of a program, so that we're not on the back end trying to really figure it out. And we've even seen, I'll be quite honest, where we've given quite specific CMC advice, and then the clinical program sort of moves along, and the CMC advice is not followed along that route, and then we have this wonderful clinical data at the end, and we still have all of these CMC pieces that are not there. And so, again, we're here, and we really do value the partnership and really want to get things to the finish line. But I think earlier conversations are. That's a long-winded answer, sorry, but.
Thank you. Holly, your thoughts on the current situation?
Yeah, well, it's so inspiring, right, to hear that the science is not what's holding us back, and trying to find new models to support this work. And I think it points to a couple of different stakeholders, right? So we've just heard about the role of FDA, engagement, and regulatory policy. There's also, new roles for academic medical centers to think about, new collaborations with industry. How long are they going to hold on to products before they transition them to a commercial model? And I think that's something that probably hasn't been explored as much as it could be. And the other thing that kind of strikes me when we talk about flexibility in this space is that we are talking predominantly around flexibility regarding manufacturing standards. Often when I think about regulatory flexibility, or when FDA is being pushed towards regulatory flexibility, it's when there's not great evidence to support the science. And that's not what we're seeing here, right? You're giving great examples where patients are doing remarkably well, and we need to find ways to get those products to patients more quickly. And I think it's just a different type of flexibility than we're used to talking about.
Mm-hmm. Great. Lee, anything from you?
Yeah, so my perspective was from my previous role as Chief Medical Officer of CMS, and really almost three buckets. There's the Medicare, there's the commercial, and the Medicaid and the pediatric population. And from CMS, they can only pay for things that are reasonable and necessary for the diagnosis and treatment, so we need FDA to approve it, and then CMS, which has a different standard, can pay for it in the appropriate setting, and that's usually academic. So one of the problems is these have all been studied in AMCs. They're not really disseminated out. Now, I just came back with Ellen from Milken, and we heard about great technologies that may be occurring that could be done in any place that has a blood bank. Hopefully that is the future, as opposed to needing a transplant center. But so the first would be, how do you get Medicare coverage? When you talk about commercial, and I was at AHIP last year, the costs of these are very large, and they all look at their 2-year time horizon. Even the self-insured employers in today's world worry about the 2-year horizon of people switching to another employer. So do you have subscription plans? How do you pay for this where it's not a population-based approach in which you have a long-term outcome? And in the Medicaid population, and I learned a lot, my cousin Gary Fleischer is a physician-in-chief at Boston Children's. Each state is so different of what they will cover, and the problem is when you have Stanford or Boston Children's, Philadelphia, CHOP, where we are, what if you're not in a state that has a major pediatric population, but you need to send them there for the treatment? How do you figure out a payment mechanism that crosses state lines that actually is associated with different states? So I think the positive is that I think it was 43 states signed up for the cell and gene therapy CMS, CMMI model to sort of share the costs, but that's only in pieces of it, and we still don't know the details yet.
Thank you, Lee. Kirsten, any thoughts on your?
Yeah, I think we need to get creative about the entire drug development model. The biopharma model, biotech, moving on to pharma, works well for products that might ultimately be profitable. But it's very hard to expect a publicly traded company to invest in a product where there is never going to be a path to profitability. So how do you incentivize some alternative development path? And it may not be the biopharma model. It may be bringing in the other stakeholders, the payers. Maybe the payers can provide payment, at least cost recovery for drug development in these rare diseases. The diseases are rare. They're not going to break the bank of the payers. So I just think we need to get very creative about thinking of alternative pathways other than for-profit biopharma companies to develop these therapies for rare diseases when it's evidence early on that they really work. I mean, when you have a breakthrough therapy, like for pediatric ALL or some of the CAR T programs that I worked on in blood cancers, you know in the first few cohorts of a phase 1 trial that this is a breakthrough therapy. This is going to change the treatment landscape for this disease. The FDA knows that it's a breakthrough therapy. They grant you breakthrough therapy designation. You know, how might there be a way to get cost recovery earlier so that maybe a more philanthropic organization can take over the development? Maybe an academic medical center can afford to take over the development. I just think we need alternative pathways. Otherwise, there's a bar. And diseases below that bar will never be developed.
I might, yeah, a couple.
Well, I just wanted to add to that. I think we have to be more intentional about moving things earlier into sort of a more non-competitive space and information sharing. You know, we often hear at FDA, well, okay, can you just share with me what someone else did, you know, for this? And obviously, you know, that's not something that FDA can necessarily do, but I do think that in some of these rare disease models where, you know, I agree with you, it's not sustainable to have multiple people developing things that affect maybe 10, 15 patients in all of the United States. How can we get to a place where we're using more platform-like approaches where we are sharing information across those types of programs? We're sharing some of the, you know, the preclinical lessons learned. We're sharing that information. You know, at FDA, we are very comfortable and in a space where we allow leverage across programs if it makes sense and if the you know, the backbones are the same, and otherwise there's a lot of flexibility that we do have, but I think that there needs to be more information sharing in a non-competitive space.
Crystal, you had something to add?
No, I appreciate that, Nicole, and you know, some of that's on, I think the FDA can't do that for us.
Right.
We have to do that, and that's something, you know, we need to take to our community. I think the community is aligned to, you know, do what it takes. I want to comment on a couple of things that Kristen said, because you know, we've recently launched a nonprofit, Access for Kids, to try to bring the GD2 CAR and a couple other assets, you know, to FDA approval. And I'm learning a lot about the creative models and what will get traction and what won't. And the first thing that we need to kind of understand, and Lee, you can kind of confirm this, and Holly, that anything short of FDA approval will not solve our problem. The cost recovery model, when I first learned about that 5 years ago, that sounded like, you know, this was the answer to our prayers. We could simply bill the payers for the manufacturing costs for the drug we're making at the academic medical center, and we can continue to deliver it without these skyrocketing clinical trial costs. Turns out that if there isn't FDA approval, none of the payers will pay. Full stop. So cost recovery, while in theory sounds good, in practice, it does not solve the problem. Number two.
Can I just say a little
yeah,
bit? I just
yeah.
want to make sure people know what you mean
Please,
when you say
go for it,
cost
Holly.
recovery, right?
Yeah.
And Nicole, obviously, you should speak to this as well, right? But the idea is that you can charge for investigational products. Typically, that's not how things work, but the cost recovery is limited. You can't profit,
Nope,
and
right.
there are certain things that are not allowed to be covered, right? So you can charge for the direct costs of making the drug available, and that doesn't cover administration costs. I mean, administering to the patient on top of administrative costs, right? So there's lots of things that are not covered. It does make sense, though, that payers won't pay for things until they have FDA approval. This is an exceptional case where it's so frustrating, right? It feels challenging. Why can't we get the payers to pay for something that works? But the broader rule makes a lot of sense, right? That payers come onto the picture once we've figured out that a product is safe and effective as designated by FDA approval. And part of my role, I think, on this working group and the panel is kind of to remind us that exceptional cases come up, but if we apply the same rules in those exceptional cases to the common cases, we're going to find ourselves in trouble. So if we say, well, it's okay to pay for products before they get FDA approval, then you've got other people who come along and say, well, we think our product is promising. Payers, why won't you pay for it? What do we mean by promising? You've got data on cures. Not a lot of companies, not a lot of developers have that strong data. And so I think we do need to come up with exceptional approaches for these exceptional cases while making sure that we keep tight constraints for the more common.
But there are statutory limitations to that. Medicare and Medicaid can't. There is a clinical trials protocol that it can pay for everything except the manufacturing, so it can work. In fact, it's interesting, you can actually, if you have an NIH protocol, you can get payment from Medicare and Medicaid if it's approved by the agency, but if it's PCORI, you actually can't. But that's in statute, so you'd actually have to get congressional action to change some of these things, at least for federal payers.
I do think that we're supposed to be a panel thinking of out-of-the-box solutions up to and including congressional statutes.
Yes.
But there, you can build off of mechanisms that already exist. We have the breakthrough therapy designation. That identifies products that are promising early, usually in phase 1, sometimes after 5 or 7 patients. Why not combine that with a numbers threshold? If your disease indication is below a certain number, defining it as rare, and you have breakthrough therapy designation, now the payers have to pay.
But that takes Congress.
Well, let's get Congress to act.
And that gets scored by CBO, which means it will affect reconciliation, which means, Marlene and Ellen, that's not going to happen right now. But yes, perhaps after we get reconciliation, we can go back to Congress.
Rather than kind of blowing up the system in which FDA approves and then payers pay, if there's clear evidence that a product is beneficial, then we've got a clear path, right? Then
Accelerated approval is fine.
FDA can approve, right? And then payers can pay.
Yeah.
So I'm not sure we need to go to Congress.
Well, I think you can take many examples for CAR T cells where the breakthrough therapy designation comes years and arguably hundreds of millions of dollars before the accelerated approval. The accelerated approval still requires a substantial additional body of clinical and CMC data beyond the breakthrough therapy designation. So if we want to get some other payer engaged, I do think there's a huge gap between accelerated approval and breakthrough therapy designation that we could tap into.
But we could change that too. We could actually change FDA authorities that match payer authorities, which is the other solution. Sorry, we've hijacked Crystal.
Well, our FDA commissioner is proposing a new pathway for therapeutics that target a molecular abnormality that could occur earlier in the drug development cycle. So there may be a path there.
I heard regulatory change, some legislative change. We also talked a little bit about incentivization. I don't quite know what that means because we didn't really talk about that, but that might be an important topic to touch on. But we're going to run out of time on this first question. But I'm happy to talk about incentivization if somebody has some thoughts in that regard.
Well, I do want to, Kristen, to your point of philanthropy, you can't sustain, drug development on philanthropy. You can launch efforts, but there's just no way that that's going to sustain things. What we can talk about are new business models that remain in the private sector that engage funders who will get a return, but it's a lower rate of return, and it's just enough profit to keep the flywheel moving. I think those are the kind of new business models that we need to develop, and in order to make those work, we need to access different pots of capital than you and I are used to accessing. It's not the venture capitalist who's going to invest here. It's the family foundation. It's the mission-related investors. And greasing that wheel and figuring out how to access that capital could go a long way to creating these private corporations who provide a return, but limited profit.
That's a great comment. That's a great comment, Kristen. Let's talk a little bit about manufacturing and GMP specifically to move on to that topic. So for clinicians and researchers, let's say, where have you seen manufacturing and CMC stall development? And that could probably take half an hour for each one of us, so let's focus it in a highlight. And how might flexibility be applied responsibly in those cases? Crystal, would you like to start off on that?
I'm going to defer to these guys first.
Kristen.
Yeah, there are so many examples.
Yeah.
I think part of the challenge is manufacturing innovation can happen generally very quickly. They're often solvable problems by engineers. You're not dealing with the fundamental challenges of cancer biology and the immune system. But you get locked into a manufacturing process early, and it's challenging to change and to do the product characterization and all the additional studies to prove that the product hasn't changed in any meaningful way. So it slows down the pace of manufacturing innovation, which is arguably the easiest way to bring the costs down. You get a CAR T-cell product approved in a small number of patients, accelerated approval, and you set your release specifications based on the data you have. And then you gather more, and those tend to be pretty narrow because you can't justify the broader specifications. And then you treat 100 or 1,000 more patients, and you can justify changing the specifications. Right now, that it's a complicated process. Well, how much data do you need?
Yeah.
To prove that if you narrow those or if you broaden those specifications, you haven't fundamentally changed the activity or safety of the product. So I don't know exactly the answer. I think to your point, if you really understand your product and you understand the characteristics that drive safety and efficacy and exposure, then you should be able to do very simple comparability studies and keep innovating the manufacturing process.
Yeah. But that's part of the problem, right, in that we really don't understand many of the attributes that contribute to the activity. So we're kind of stuck in that circle. What kind of tools do we think, technical, policy, otherwise, might increase our confidence in having that kind of flexibility without compromising when and how the flexibility is applied? Because, of course, flexibility means slippery slope, right? So how do we avoid that? Maybe, Nicole, you can start off on that.
Yeah, I mean, so first of all, the FDA, I really want to stress here, we are a partner. And I say that because there are some stalled programs, and we're not the same FDA that we were 10 years ago. We've evolved a lot in this space, and with evolution comes the ability to really sort of problem solve some of these issues. And so I say that because we have had stalled programs come back to us, have additional conversations, and we've figured out ways to get things moving again. So I really want to put that out there. Please do not hesitate to set up meetings with us and come back and talk to us. Our advice, I'm just going to be very honest, might be different than what it was 10 years ago, and we are really committed to helping in that way. Another, I think, very important program to also highlight is we have a CMC readiness pilot program. We have about 4 active programs that are in that right now, but that also allows additional touch points with us around CMC manufacturing, having some of these conversations with some of the therapies where the mechanism of action is not quite understood, and sort of what can we do early on to make sure that we're ready to launch a product. So I really think people should take advantage of some of our existing programs. And within the RMAT framework, you mentioned breakthrough framework, there are a lot of additional touch points that we are more than happy to have and to understand some of the flexibilities that are unique to individual programs. I do think we really do realize at FDA we have to right fit our advice for the size, for the population size, for if there are no other available therapies for an indication. And we hear you. We hear from patients, from advocates all the time. They're saying, I'm willing to accept that increased risk for potential benefit in an area where there is just not a lot. And we take all of those things into account. I really just wanted to stress that.
Yeah, thank you.
It's important.
I had one comment I wanted to add. You talked about making more information available.
Yeah.
I think, there are regulations around, let's say, cell and gene therapy, and they're often general, appropriately so, and you comment the FDA evolves as the field evolves, but most sponsors aren't aware of all the feedback you've given to the hundreds of other applicants, and so that they can stay current with the evolution. And so they revert back to the 10 years ago when they last had this conversation with the FDA, and everybody's very conservative. So if there's a way that those case studies can be shared, de-identified, so that everybody is at the state of the art of how regulations are being interpreted and the nuances around the size of the indication, et cetera, maybe product characterization, because there's a lot of convergent biology. We think it's complicated, but if you look at all 100 cell therapy products, I'm sure there's a lot of convergent biology around product characterization that predicts clinical outcomes. Is there a way that we can leverage that information sharing so that all those next-gen companies aren't reinventing the wheel and spending money unnecessarily?
It's a great point, Kristen. Lee, you had something to add to that?
Well, there's at least the 3 years I was at the agency, and I believe it continues to have a lot of discourse between FDA and CMS. And if you look in the device side, there's minimal evidence, and yet we approve 510K and de novo products. But we also sometimes continue to collect evidence, both post-marketing, and as we're going into these areas, that may become a more and more important process, because these would probably be Part B drugs. They're probably intravenous given, in settings. And I'd be curious, Holly, because there's been a lot of question of should we require registries to make sure things that we're not sure if we believe it works, but we can't guarantee it works. And to be honest, we should continue to evaluate whether it works, especially as newer technologies come on board that may surpass it. But are the patient population who are receiving this willing to be part of registries so that we learn rather than waiting?
Yeah, just to comment on this idea of registries, right? Remember, I'm a lawyer. I'm not a statistician or a
But
trial
you're an
designer,
ethicist.
but I'm an ethicist. So the thing here's what I'll say about registries. Definitely better to have them than not have them. What makes me worried is when we talk about things like the Promising Pathway Act or this new kind of rare disease pathway that's been floated, and the idea that maybe registries can substitute for other more rigorous ways of knowing. And I think they all have a role to play. I get nervous when it comes to substitution, but that's not what you're describing. And if there are cures, right, if things that you can kind of see without controlled designs, then registries are fabulous for that. And I think that is something that payers have a mechanism to pay for. The other thing, just to go back a moment, you've inspired me. We should be thinking outside the box here. So much of what we think about in this space and how FDA regulates has to do with kind of traditional sponsors who have profit motive, and they don't want to necessarily share information with each other. But part of what you're describing, you didn't use the word nonprofit. I don't know that that's exactly what you had in mind, but there's philanthropic approaches, near nonprofit approaches where you could imagine consortia coming together of collaborators who say we're willing to share information with each other even though we're not setting up a company necessarily. And there might be things that could change in both regulation and statute that would facilitate greater sharing of information kind of on an opt-in basis through entities that don't have the same kind of profit motives that have run in pharma and biotech in the past.
Even for the profit motivated, there must be some incentivization method that you get these folks to do what share data, and then you get this sort of thing, right? Kristen.
Well, let me ask Kristen, do you think that on an anonymized basis, because so much of this is in pharma, so much of this knowledge, this evolving knowledge is in pharma. I mean, we academics can share, but I'm not sure that would be as impactful as what the private sector has. Would there be appetite for that or not?
I mean, I think there would be. I've heard Nicole share case studies, super informative case studies around various accelerated pathways or CMC feedback, de-identified, that would be really useful if every next-gen startup cell and gene therapy company were aware of how the FDA had responded to that last IND question, starting dose justification for one example. I do think that pharma would be more flexible because they felt the pain of having, regulations that aren't entirely clear, things that have slowed them down and stopped the pace of innovation, that there's a certain amount of sharing that would, be the tide that raises all boats that pharma would be willing to do. So I think it's about getting the right stakeholders together and say, what can we share broadly so that this whole field moves forward faster, including the products and the innovation happening in pharma. Yeah.
Kristen, do you think that pharma is more concerned about data or regulatory strategy or both?
I mean, there's intellectual property.
Yeah.
But there's also the cost of development and the path to profitability, as my colleague has instructed me on. And there has to be a path. And if you can speed up the cost of the timeline of development, if you can lower the cost of development, you can create a path to profitability. Yeah. And that includes all of these things we've talked about: data sharing, appropriate regulatory policy, maybe some out-of-the-box ideas around cost recovery.
Yeah. Yeah. Okay, great. Thank you. Let's move on to the topic of access and benefit risk. And we touched on what makes it hard for payers to support access today. So I don't think we need to touch on that anymore. But from an industry perspective, perhaps it might be useful to understand what kind of clarity or incentives would help companies pursue smaller, less commercially viable populations. And Kristen, maybe you can kick us off on that.
Faster path to reimbursement. Mm-hmm. So, if you can lower the cost of development, you might be able to incentivize products that are going to overall have lower profitability. Because it's how much do you have to spend before you ever have a single penny of revenue on a product. If you can lower that cost, I would hope, you might lower the threshold of when a disease becomes, and a patient population becomes interesting.
Mm-hmm. Lee, you have some thoughts on that?
Well, I'm actually curious. Big Pharma, do they give earlier access to their own employees for this, or do they actually demand? It's an interesting, because my question is, we're taxpayers, so that's Medicare and Medicaid. They also have profit motivations in the insurance industry, certainly, and they're trying to ensure that they provide the right care, but they're using a standard that's been developed, which is the FDA standard. So I'm curious how we balance that. A lot of places are self-insured, and that's what I'm hearing when I'm at AHIP or others. I'm hearing, do we have subscription models that allow individual, entities to decide we want to provide these earlier access to some of these things, like gene therapy? I
mean, there are standard regulatory pathways for early access, either early access just in the peri-approval window or single patient IND type initiatives. That's very hard to invoke for cell and gene therapy when you have limited manufacturing slots that are being used to enroll the patients in the pivotal trial to lead to approval, for example. No, companies don't say we're going to provide early access just to our employees. You either have an early access program or you don't. If you have it, anybody who's eligible is eligible.
Nicole, any thoughts from you on this topic? How do we incentivize companies to pursue less commercially viable opportunities?
I think it's a really complex problem, to be honest. At FDA, we are really trying to do our part to everybody wants, obviously, access to therapies and approvals earlier. And I do think there is a lot of opportunity in the, cell and gene therapy space for that, because we have more targeted therapies. We can, based on that target, and we see robust effects or an effect on a particular mutation that we know exists, and then I think that we can get to earlier approvals and therapies than what traditionally we've seen. And I think in particular in the rare disease space, there's a lot of opportunity there. We don't have to necessarily wait, 10 years to see a clinical effect. And so, again, I think I don't have the actual answer here, but I do think at FDA we have a lot of tools that we are willing to use to embrace these pieces to hopefully incentivize the continued development of rare disease programs where there's not a lot of patients.
Great. Crystal, you have something?
Profitability, yeah.
To that point, I wonder, Nicole, if you would be willing to speak on the platform designation, which recently was rolled out, and what, where it stands, and.
Yeah, the designation itself, it does require that there be an approved therapy, but what we have done in our office is move well beyond that. We have really embraced sponsors that are even prior to having an approved therapy using these platform-like approaches. And so we have, for example, we have backbones that are being used across 6, 7 different indications, and we're leveraging information in that non-clinical space, in the CMC space, such that it does hopefully overall expedite development for the individual indications. We're leveraging information in the clinical space too, which goes a little bit beyond, I think, what was initially the intent. So, using safety databases across several different indications to support an individual indication. And so there is a lot of opportunity, and we have seen sponsors using it quite a bit more in the past year, and encourage that as well.
Great.
I'd add one comment on sort of a platform. A real-world example of that for T-cell therapies is TCR-based therapies, where a given TCR is going to recognize a peptide in the context of a given HLA type. So, of course, everybody develops their TCRs to the most common HLA type, AO2. And now you've proven it works, you even have it approved. First genetically modified T-cell therapy for a solid tumor is a TCR-based therapy against MAGE-A4 HLA-O2. How do you expedite the development of MAGE-A4 targeted TCRs against other HLA types? It's a platform. And if you have to go through the whole clinical development program and the CMC and another hundreds of millions of dollars to move that, in a sense, almost the same product to patients with alternative HLA types, there has to be a faster way to do that. Otherwise, you'll never develop the product for the other HLAs, and there's so many patients that could benefit who will miss out.
And we completely agree, and I would say come talk to us.
But is that the role for post-approval evidence collection? Because it doesn't guarantee, just because the platform works for one HLA doesn't guarantee, it certainly strongly suggests, and that's what I've heard. The previous administration was very focused on post-marketing. I've heard Marty say he's interested in post-marketing. Is that the trade-off that we're willing to take?
We certainly are willing to take it. Believe me, these children are followed for decades.
Yes.
We will do post-marketing monitoring,
That's great.
to the hill.
Let's talk a little bit about the benefit-risk trade-offs in serious conditions where no therapies exist. So in high-need conditions, patients may be willing to accept more uncertainty, as Crystal just talked about here. Now, how should this influence benefit-risk decisions in the application of the regulatory flexibility we've been talking about a little bit? What frameworks might help guide the responsible use always in these scenarios? Holly, maybe kick this off for us.
Well, look, I mean, I don't think it's a surprise to anyone. We've heard on this panel already, it makes all the sense in the world that individual patients and their families would make trade-offs when it comes to risk-benefit and when it comes to uncertainty. And I think FDA has shown tremendous flexibility, in accepting those trade-offs. The part where I would kind of offer some caution is recognizing that while it might be reasonable for individual patients and families to make those judgment calls, FDA is a public health agency and has to make decisions that apply, more broadly than that. And so a patient might say, or a parent of a child might say, whatever you've got right now, give it to me, right? And FDA can't say that because we actually do need to figure out whether products are safe and effective, or we'll find ourselves in the same position 5 years down the road, 10 years down the road, not really knowing what works. And the patients are then going to face the exact same choice. So FDA standards, of course, are and have to be stronger than what any individual person would be willing to do. But they've shown, tremendous flexibility. I've been a critic, right, sometimes of, too much flexibility. But again, to come back to a point I was making earlier, what we're talking about here is a completely different type of flexibility than what we saw with regard to aducanumab or, some of these other highly criticized decisions. You've got really strong evidence early on of massive benefit for these patients. And that's why I'm pushing to say, well, let's just get the FDA approval rather than, in a traditional way, rather than saying, let's do backflips to figure out how payers can pay for things before FDA approves.
Makes sense. Crystal, some thoughts on that?
Agree.
Crystal, some thoughts
Kristen.
on
We keep, I'll answer.
It's hard,
Kristen.
that? Going off script. Yeah.
Yeah, I spent most of my career running phase 1 cancer trials, where you have no evidence of benefit other than perhaps in animal models. And in the context of informed consent, patients can choose to be in a clinical trial to receive that investigational therapy. So there are models to receive therapies where there is a lot of risk and benefit is unproven, or a lot of potential risk. The question is, the whole burden of payment for that model is on the sponsor, is on the biopharma industry. If there is a way to have some other type of approval at much earlier in development such that the payers start paying, you can always give a consent form to the patient with the rare disease, say, this is what we know, these are the benefits, these are the risks, do you want to participate? Like, why not treat the semi-approved product that's reimbursed by another stakeholder with the same kind of informed consent we treat patients who participate in clinical trials?
Yeah.
Well, I know. I was going to just answer, why not treat it the same way? And I think part of the reason is, it's one thing to say for, ultra rare and rare disease, right, that will have a lower standard and it will just be up to you to consent. But there are many patients for, bigger diseases who would also say we're willing to take that risk. And then you kind of go down the chain and say, well, what do we need FDA for at all, right? Or we're kind of like back to the Laetrile standard of saying people are willing to try it, and so why don't we let them, and why don't we have the payers pay for it? I mean, I do think we need to have a regulatory standard, and where that standard should fall along the spectrum, I think is perfectly reasonable, up for debate. But I think we need FDA, right? I think we need standards before we kind of throw it out to the wind and allow people to try anything.
And sorry.
No,
go
please,
ahead.
go.
And when CMS was initially looking at the cell and gene therapy, I was still in the agency, and the question is, how do we price it if you give somebody earlier approval, getting to Holly's point, whatever we come up with? And the question is, how do you give a discount, and if it works? We couldn't figure out how to actually do that mechanistically, and then should we cap pharma's profit margin at the end on these drugs if it works? I mean, in some ways, in our system, who should be funding the innovation? I agree with you entirely. For ultra rare diseases, we need a much better mechanism, and we still need the FDA to say you've reached that bar. And it may not be randomized trials. 100% agree with that.
And I'll just add, another issue that we think about too is, how do we continue to foster innovation in this space with the cost models that we have, where things are quite expensive after an approval or otherwise. But we really want to continue to encourage and foster innovation in a space with, in rare disease spaces where there aren't a lot of patients. We have to continue to adapt and to encourage that. And I think that's an added problem that we don't have necessarily a solution to. But yeah.
Especially with the NIH cuts, which is making it more difficult to wonder, because so much the early work comes out of academia.
I'd comment, we have a model. Okay, so you go first,
I want to echo Holly's call that what we need is nothing short than FDA approval, full stop. Because there are two sides to doing it with a consent form. Number one is all children who need it will not have access. It will only be those children whose families get themselves to the tertiary care center and understand clinical research. So we will not solve the problem of access. We'll also have the flip side. Believe you me, patients, families who have children who have dread disease are extremely vulnerable. And lowering that standard to giving a pass to things that really aren't effective opens the door to a whole host of people who will market drugs that don't work for these children, and it distracts and it takes up precious time, and they don't get the drug that works. So I really am a firm believer we need FDA approval, full stop.
Well said, well said.
That's a great point.
I
No,
agree.
I mean, I agree with that. The one comment I'd make is, we have a system, a regulatory system, a biopharma development system that works for certain diseases above a certain frequency size. And then we have the examples you mentioned of the products that are not getting developed at all, and people with those diseases are dying. So the risk of not doing something is very, very high.
Yeah.
And so whether it's giving full approval based on less data, whether it's not requiring 3 PQP runs, CMC runs, whatever it is, you have to be willing to take more risk, because the alternative is there is nothing, and the risk is 100% certainty of death. So when that's the alternative, why wouldn't we take more risk?
It's a great way to end this particular session. Let's talk a little, I think we have 8 minutes to go and a couple additional topics. So let's talk about post-market monitoring. How can we use post-market data to support oversight and access for the therapies we've been talking about today? Lee, maybe you can kick this one off.
Yeah, no, I've been a believer strongly in that, and you mentioned aducanumab. That was the conceptual model that at CMS we came up with, which is this idea of continuing to learn afterwards. It was interesting after the lecanemab, they actually did require that. They didn't in the aducanumab decision. We wouldn't have needed to at CMS to even have stood up the registry, and in fact, FDA frequently uses claims data to constantly monitor. So I think whether it's FDA, CMS, I think the ability to continue to monitor and as new therapies come along, see if it achieved the same goals in the real world is the way that FDA then can take action and take it off the market if it doesn't work. And I think that's what we heard a lot about recently.
Kristen, any thoughts from you?
Yeah, the challenge with the post-marketing requirements is that the cost of the burden of all that is all on the sponsor. So if you're trying to convince a for-profit industry to invest in rare diseases, adding on the layers of responsibilities post-marketing, cost, 15 years of follow-up is going to de-incentivize the development of these treatments for rare diseases. I mean, it's incredibly important. Why not have the payers pay? Maybe I'm sounding like a broken record.
I would just say the goal of interoperability should be to get us there. I mean, so maybe the industries can, and we're trying to talk about this a lot, if you truly have interoperability, then most of the data will be in the EHRs. You will be able to do this at low burden. So rather than industry pushing against this, we should all push towards interoperability and have the FDA be able to analyze it. They're the scientists in this space, and people like Holly and the academic community look at those data, and that was really our goal. It was only 10 data elements, which took 6 minutes, 4 times over 2 years. That was our vision for the Aduhelm registry. It was really low burden because it was all going to be done with claims data.
Any other thoughts, Kristen?
No, I don't have anything to say.
I mean, I would say we need to right fit post-marketing. So, what we're looking at FDA is, so for example, across, certain classes, or if we have multiple indications that are now approved for an individual product, how can we sort of, right fit it such that it makes sense, and we're not requiring, significantly burdensome types of post-market programs. So I think that is definitely a conversation that we need to continue. But post-market commitments and requirements are really a very strong piece of this as we're talking about getting things to patients earlier. We really need a commitment in the post-market setting to really do this right, to make sure that we are capturing information. And we've seen some variability there. And then, it's just challenging. As you mentioned, some of the more innovative therapies are in an academic setting. And then, for example, we've had real life problems where that investigator leaves that institution, and then no one wants to take up the long-term follow-up piece, and sort of what do we do? Because we have several patients out there that really need follow-up and additional touch points. And so there's more that we could have a whole session on post-marketing. I think it's extremely important, but we're going to have to sort of think through it a bit more.
And this 15-year follow-up, which is sort of this gorilla in every conversation that we have, doesn't help things at all, right? I mean,
Agreed.
Do we really need that for cell therapists? Yeah. We have less than 5 minutes, and a final question to everybody here on the path forward. So what would be a meaningful and feasible next step, right? Guidance, pilot programs, demonstration project, to advance the regulatory pathways to support access to the rare cell therapy applications that we've been talking about. Chris, I'd like to start with you.
It's wonderful that the FDA offers time with developers of, products for small markets, and I agree with you. I think that really is key. But I think there is also value in putting out some of these ideas in print that basically say we have flexibility around manufacturing. Decentralized manufacturing, small batch manufacturing may not have the same commercial requirements as large batch. Because the problem is, as we're trying to raise money to, basically get funding to move these therapies forward, the investors need to know that the regulatory risk is really reasonable. And having some evidence in paper to the extent that maintains the flexibility that FDA needs, that would be very, very helpful, I think, for our field.
Thank you. Lee, your thoughts?
I agree with Holly. Actually, I think the FDA, and I really want to thank, have approval, but use all your flexibilities to actually approve it earlier. Or Congress should give more flexibility, but make it an approval process, not a pseudo-approval process. And then I strongly believe in interoperability and post-marketing fit for purpose.
Great. Holly?
Well, we've had kind of an apolitical conversation so far, and I'll just dip my toe into the political waters and say everything we've talked about requires deep engagement with FDA, and if you don't have FDA staff who can meet with you, then all of this is going to fall flat. So making sure that the agency is well funded, well supported with staff, I think is point number 1, 2, and 3. And then I'm going to throw out your feasible criterion and just say, if we're thinking outside the box, we've talked on a number of these questions about the role of payers. Obviously, there are private payers and government payers. But if the government is paying, through post-market coverage or registries or paying for early access or those types of things, why not also think about novel federal funding mechanisms that are different from insurance coverage? Bridge funding, things to kind of get products developed in this non-commercial model. I'm especially in the context of rare disease, often thinking about what's the responsibility of the government to meet these gaps where the capitalist system and commercial sponsorship is not going to work, right? It is a responsibility of the government to step in those cases, and there might be novel funding opportunities to do that without relying on insurance payment.
ARBA H.
Yeah.
Kristen, any final thoughts?
Yeah, I think the focus is how do we develop these complex but incredibly beneficial cell and gene therapies for rare disease, and I think it is the two things we've discussed. You need a regulatory pathway that lowers the cost and time of development, 2, some type of approval that triggers payers to pay. And then we need new capital investment from a different source. It can't just be the biopharma industry and venture capital. And the obvious source is the payers, whether it's the private payers or the government payers. I think we need a different source of capital, which means society needs to decide that this is worth paying for.
Great. And Nicole, the final word on the final topic, the FDA represents.
So I have been listening to a lot, and I agree with everything that that has been said. I think there's opportunities for, we have a lot of guidance out there, but to really sort of consolidate that into one to address what we're talking about, I think that there's some opportunity there. And phase-based approaches, we have a lot of conversation about like what phase are you in your program and what do you really need for that phase. We have to communicate that a bit more. I think that we see a lot of variation in terms of sophistication of sponsors, which is great. We have very small firms, academics, and large companies, and there really is the gamut of understanding of that. And so I do think that at FDA, we can continue the momentum. We've had a lot of success, I would say, over the past couple years, and really trying to hopefully right fit some of these things and be a little bit more communicative, and we're really committed to that. And I just want to continue that momentum, and I'm committed to doing so. So thank you for
Thank you.
that.
Are we taking questions now, folks? Or?
Did Ellen have a question? It's on the screen. Ellen had a
Oh,
question.
on the screen.
Ellen had a question.
Oh, her question.
Ellen Segal. So Ellen, do I read it? With given political climate asking for more evidence and critical of expedited programs, how perfect does the evidence need to be? Somebody want to take that?
Well, go ahead.
Well, but I do think that we don't yet know what the administration is, how they're going to fall down on this, because I agree we're hearing about more evidence, and maybe this is around, common diseases and targeted therapies where you have an extension in PFS by 3 months or something like that. But I've also heard very clear messaging from the Trump administration that they are interested in rare disease. They're very interested in pediatric cancer in particular. Commissioner Macri has made this foray around rare disease. So I think I remain optimistic that we might be able to find a path forward with the new administration to help some of these issues.
I heard something interesting between the two. You keep, Kristen, you keep using payers, but payers have certain standards, and Holly said that, but Holly, you said something interesting about another source, and I mentioned ARPA-H, or should we have for the rare disease community, whether it's a government source, but that doesn't change the payers' roles. It doesn't, if society should fund this and work with FDA and work with Congress, should there be this alternative pathway developed with FDA that once it reaches the traditional requirement for payers, they pay, but there's something new, if we're really thinking out of the box, that's created?
I do think that in contradistinction to optimism around regulatory pathways for rare disease, we've seen, a gutting of our funding for basic science and research. And so I do worry that these federal funds that are potentially something we could reach out to here, the research establishment is going to need that going forward with what's happening at NIH.
And I would just say, in the vein of discussing additional pathways, I really just, in the unknown, I mean, there's a lot of unknown in terms of what that looks like. We do have a lot of regulatory flexibilities currently. So I just want to say that in the rare disease space that we are very much willing to use. And so, yes.
Folks in the audience, there's a QR code on the printed agenda. If you're interested in submitting a question, please access and submit the question through that code. Question from Tony Z, nurse practitioner. The discussion here today is mostly focused on autologous CAR T. What's our viewpoint on allogeneic CAR T to address patient access challenges seen currently with the autologous CAR Ts? Somebody want to take that? Allo,
Dr. Heggie?
any differences?
Okay, I mean, I don't know that it matters as much whether it's autologous or allogeneic. The question is, how much evidence do you need, and what are the pathways to approval for an indication for which the current model isn't working? So I think many of the discussions we've had could apply to autologous CAR T, allogeneic CAR T, in vivo CAR T, iPSC-derived CAR T, you name it. It's just that we have a model that doesn't work for diseases below a certain frequency. And so how do we create a new model?
Cost of time and costs of goods are considerations and benefits for the allogeneic, but they don't affect most of the questions that we're talking about here today, right? Okay.
But will that change with the cost of goods going down? I mean, that's what we heard at Milken, is that we can get down to quite inexpensive.
Yeah. I mean, it's already coming down. I think some of the challenges that pharma has is, they can't always incorporate the cost savings without incurring a lot more work to show comparability, which is another issue. But we are definitely seeing the costs come down.
Yeah. Question from Zina Manji. With panel 1 discussion, flexibility, and registries, is there room for pre-approval evidence and some shifts to post-approval evidence, especially in exceptional cases for more conditional approvals? Holly?
Yes, right. I mean, yes, FDA is already doing this. I think they've kind of pushed the envelope, I think, in many of these areas of flexibility, trying to figure out where along the spectrum approval should come in terms of how much evidence you generate. The thing that worries me about post-approval evidence generation typically does not worry me in these circumstances. So often, in larger diseases where you have some evidence that it's promising, but you don't have a validated surrogate, for example, and then all of a sudden the product gets accelerated approval, everyone can access it, right? Their doctor will write them a prescription, their insurer will pay for it, but it's a disease or a level of benefit that you still really need randomized trials. Those become very hard to do once everybody can access the product. That's not the scenario that we're talking about here. So this is more just about kind of long-term follow-up to evaluate durability of benefit and those types of things. So I'm more confident about our ability to rely on post-market evidence generation in the kinds of scenarios that we're talking about today than in kind of more traditional small molecule accelerated approvals.
I have a question from Steve Feldman that I can't read it fully. Can non-approved rare disease platforms leverage platform technology from approved products, reducing the PPQ and cost, and tie RMAT to a pre-approved and cost recovery process? Maybe that's you again, Holly? Maybe somebody
I'm
else.
following the question. I'm trying to read it here.
Can non-approved rare disease platforms leverage platform technologies from approved products, and in that way
Maybe a
reducing
Nicole question?
PPQs?
Yeah, I
Maybe-
think that's a Nicole question.
Nicole, it is a Nicole question.
Yes. So the platform designation itself is an approved product, and then subsequent products or subsequent indications or otherwise are leveraging the information from the approved product. And yes, I do think that affects the cost models and hopefully will bring costs down. And so there's several different platform types of models that people can leverage information, but certainly you can do that.
And when we say platform technology, what does FDA mean by it? How does FDA interpret that?
So platform approaches and platform technology can mean a lot of different things across different, across the pharmacology, toxicology space, across the CMC space. The technology itself, for example, could be, there's so many different examples, but same AAV backbone, but using a different transgene, that would be a platform type of approach or technology. So we have a lot of different ways that people are using that. It's sort of a loaded question in that regard, but platform approaches is what I would say more than the technology itself.
Great, thank you very much. I think we're at time, so I want to thank the panel for their excellent input on this. And we're wrapping up and going to Jeff's transition for lunch, correct?
Thank you.
Thank you, Michael, and thanks to all of you for a very lively discussion today. We appreciate it, and for all your insightful thoughts that are captured in the discussion today, as well as the paper. As we do transition to a break, you'll note we are using a slightly different Q&A platform today, so I do encourage everyone to go ahead and scan their QR code and submit questions that way. But with that in mind, we have embedded in an additional break here. There is lunch that will be available outside, so I don't want to hear anyone talking about going hungry today. But before we do break, I do want to just take a moment with a captive audience here to highlight a couple of projects that we have underway at Friends of Cancer Research that I hope you'll keep an eye on and consider being engaged with or passing on to your colleagues. The first is recently we launched a project called the AI RECIST project to evaluate AI-driven tumor measurement tools alongside human readers of RECIST criteria, so evaluating solid tumors for response. We've been working with 12 leading technology developers and a cross-section of experts in the field to be able to try and address three main objectives. The first, to assess AI tool agreement. Do each of the 12 different AI-based tools provide consistent tumor measurements? The second, to compare variability among AI tools and human assessment. How well do AI-driven tumor measurements align with RECIST-based readings by human expert radiologists? And third, to explore AI's impact on efficiency. Can AI tools reduce variability, maximize resources, and streamline clinical trials? By enhancing the speed and reliability of tumor measurement, AI could accelerate clinical trial progress and improve timely patient access to effective treatments. We believe that this project can help support the validation of these types of tools and set the stage for developing advanced measures for tumor burden into the future. A second exciting collaboration that I want to highlight today is the External Control Arm pilot project. This project is working to answer the question, how can we reliably leverage external data to support the evaluation of new treatments, particularly in situations where traditional randomized control trials are not feasible or ethical? The ECA project is building upon Friends' RWE portfolio to identify and advance best practices for the use of real-world data in cancer research. Through leveraging insights from our past RWD initiatives, this project is a partnership with 7 leading health data developers where we're aiming to reconstruct the control arm of a recently completed clinical trial, each of them using their own data. From this project, we'll be able to identify and evaluate how different sources of external data can replicate a target population and whether similar outcomes are observed. By constructing 7 different external control arms using different data sources, we can explore consistency in outputs, establish characteristics for fit-for-purpose data sets, and identify opportunities for using ECAs to augment single-arm clinical trials as appropriate moving forward. So now, I thank you for allowing me time for my infomercial, and in recognition of that, we will now take a break. Again, there's lunch outside. In the foyer, please feel free to visit, network with your colleagues. You're welcome to bring food back into this room. In about 40 minutes, around 12:10, we will move into our lunch keynote discussions. All right, if everyone could grab seats, I think we will get started with our afternoon sessions. Well, as everyone is getting settled, I will just take 1 minute to highlight. I hope that you've seen some of the rotating slides that we've had up to provide some more information about upcoming meetings and projects. In order to get more information about our 3 upcoming meetings over the next couple of months, please visit our website where registration should be open. We hope that you will consider joining us either in person or virtually for any of these events. But without further ado, I would like to introduce our keynote conversation, which will be moderated by Provision Policy's Kate Rawson. Kate, I certainly want to thank you for your insights and steadfast reporting over the years, and for you and your colleagues' continued partnership on numerous events with us throughout the year, and thank you for all you do. So I would like to bring you all up to the stage, please. I'll welcome Kate and the group here. to introduce the timely discussion that they've prepared for us and great participants that we have joining us today to hear from some future-looking opportunities as we think about next steps into the future. So thank you very much, Kate.
All right, great. Thank you, Jeff, so much for that introduction. It's always really nice to be here with the friends. So we are your lunchtime entertainment, and we're happy to be here to really, I think, an opportunity to reflect on what we've already heard, continue the conversation, and perhaps bring in some new ideas and expand on what's already been discussed. We only have 30 minutes, so I'm going to really quickly introduce everybody, and then we're going to get right down into what I hope will be as engaging a discussion as we've already heard this morning. So next to me is George Eastwood from the Emily Whitehead Foundation. Sar Jill is from the University of Pennsylvania, and down at the far end there is Linnell Hoke from Bristol Myers Squibb. So a lot of very expert panel, a lot of great perspectives, and I'm excited about our discussion. So George, I'm going to start with you and go down the line. And I'm hoping that, before we start to talk about some of the challenges and maybe some of the solutions to developing cell and gene therapies in this space, let's talk about some of the potential. So if you could start, and then I'll ask your fellow panelists to chime in as well, looking ahead to the next 5 years, right? What breakthroughs or developments are you most optimistic about in the cell and gene therapy space?
Yeah, I think scientifically, there's so much opportunity to deliver on access, but I think most important, and we touched upon it in the last panel, realizing some of the commercial success to empower some of that, especially given the current climate from funding and science. We mentioned allogeneic therapy. I actually came from the cell manufacturing and donor side of that prior to joining the foundation. So scientifically, we have ways to go there. In vivo engineering, of course. I know Dr. Gill does a lot of work there. So scientifically, there's so much promise. But if we don't deliver on some of the commercial side and some of those unique models that we touched upon and talked about, it'll be really hard to deliver to the number of patients that we need to.
Right.
So we'd love to see the innovation happening on the policy side. And I think gatherings like this allow us to break apart some of the policy issues. I think the last panel mentioned, really you're looking at clinical data, clinical outcomes when you're thinking about easing and flexibility and regulatory. We have the whole side of CMC, of platform, of all of that to detangle, build the framework for, and discuss and connect in a way that we haven't before, from the manufacturing to the commercial delivery. So excited for the innovation there, but obviously many stakeholders needed to pull together in rooms like this to drive it forward.
Yeah. Sar, what can you add to that?
Yeah, thanks, Kate. So Monday and Tuesday of this week, the National Cancer Institute held a workshop on in vivo engineering and in vivo editing. And I think if your question was, what are we looking forward to seeing in the next 5 years? I think that's what we're looking forward to. And I think there are some early sort of green shoots of evidence that this might actually work. Maybe the way we ought to look at things is that there's very clear proof of concept as manifested by the presence of regulatory approval and commercial availability of ex vivo CAR T-cell therapies. And while there are major technical and scientific challenges to resolve in the in vivo engineering and editing space, it does seem to me as though if we regard the proof of concept in ex vivo therapeutics just as that proof of concept, and that now we ought to make it a bit less like a cottage industry whereby each patient has their own product manufactured for them. And I know this doesn't apply to allo per se, because that isn't the case, but it maybe it's time for us to think of the next 5 years as moving beyond the cottage industry, very high quality cottage industry, to a more industrialized process whereby the gene therapy vector is the drug as opposed to the cells being the drug, because it would undoubtedly reduce the cost of goods and increase the throughput, and presumably, probably really increase the availability and access of these therapies. But again, there are major scientific and technical challenges to resolve before that reaches the same level that we have now, the same level of comfort that we have now with ex vivo therapies. But that's what I think we ought to look for in the next 5 years.
Yeah. Linnell, what we'd love to hear what you have to think about the next 5 years.
Yeah, so I think about it in segments, because I think about the next 5 years when I think about kind of the near-term pipeline and what's about to emerge. You're going to see additional disease areas being able to be treated by CAR T. And obviously, I know this is Friends of Cancer, but obviously one of the most exciting areas now we see is the promise of what a CD19 CAR T can deliver to B-cell mediated autoimmune disorders. So diseases that once never had an endpoint being considered around treatment-free remission, now where this could potentially possible, where you induce an immune reset, and to be honest with you, kind of reset the hope and potential these patients never had before. So that's extraordinary. It's hard not to get excited about that. The other segment I think about, which is where SAR went, which is autologous cell therapy has been revolutionary. I mean, when we talk to patients that have been going through multiple cycles of different modalities as they fight different hematologic malignancies, it is always a reminder every time they go either take a pill or go for an infusion of a monoclonal antibody that they have cancer. And what the magic of autologous CAR T and CAR T in general is, with a one-time infusion, they go from being a patient to being a person again. We'll have patients tell stories where I could never go on that trip to Europe or go backpacking or hike because I was tethered to, an infusion chair. And so to me, we can't forget that there is enormous magic in that. However, we also know there's inherent complexities. And that's why only 2 in 10 patients are getting access to an autologous CAR T. Previously, it was always assumed it was because we couldn't manufacture to scale. Well, I can tell you as I sit here today, we, as well as the other manufacturers, are meeting the demand. The issue is the demand is shorter than it should be. And why is that? It's because either patients have an inability to get to a CAR T center. These are big commitments to be at a CAR T center for 4 weeks, not be able to drive for 8, and being able to only access that care at one of these refined centers. So I do think ideas of how you bring CAR T into the community is really important, and that can be done through science, in vivo strategies, HDL strategies. It can be done with autologous CAR T by creating more of an ecosystem that allows community physicians to deliver CAR T. I think we're too much bundled with autologous stem cell transplant when it comes to the safety considerations, eligibility considerations, and yet CAR Ts have been in the market since 2017, accumulated a significant amount of real-world data that would tell you that maybe actually they can be treated in the community. We see this happen today with academic centers are now using most of the CAR Ts in outpatient. Because they do believe that not every CAR T is created equal, so some CAR Ts, but a lot of them believe that these patients can be treated in the outpatient, and as long as there's tight and close monitoring, that can be achieved. But so I do find the next 5 years to be extraordinarily exciting, whether you're thinking about how the advancements will happen in these off-the-shelf solutions, as well as how I see autologous CAR T continuing to penetrate into different targets, different patient populations, et cetera.
Yeah. No, I'm excited about the next 5 years too. And I wonder, as a follow-up to that, if you could, and you've touched on some of these already just in terms of point of care and maybe, bringing the treatment outside of the big academic institutions and more into the community. What other foundational work needs to happen now in order to get where we want to be in the next 5 years?
So listen, if you look at, I'll pick a market, France. 4 out of 10 patients get access to a CAR T in France. And there is some underpinnings of that. Underpinnings are there is an ecosystem that connects academic centers and communities much more tightly. There's also economic incentives that are different than in the United States. And so I do think in order for people to get access to CAR T in the community, you've got to establish success factors for the community physicians. So being able to not only create a framework for them operationally, clinically, but also economically, so they can deliver CAR T in the community. Those are all really critical. We're starting to see that take traction. We have actual community practices today, true, pure community practices delivering CAR T today, and they're doing so successfully. So I do think broadening that, because there always will be a limit of how much patients can truly get to an academic center, and treating in the community is super important. So I do think creating an ecosystem that allows community physicians to successfully and safely deliver CAR T in the community is really important.
Yeah, great. And if anyone has anything to add, just please jump right in.
I do have a counterpoint to that, if I may, which is that I mean, access is super important, of course. Why would we do this if we don't get it to patients who need it? But I think we at least ought to consider whether delivering in the community is the right place to do it. I at least want to have a discussion about that, because with any specialized type of treatment, I think it's true that volume correlates with quality. And so, I think that in volume, there's never going to be the same volume in a small, by definition, in a small community center that there is in a large academic center. And so there is a give or take that I think is actually really important to consider, whether it is actually in the individual patient's advantage to be getting treated at a community center that may be a 10-minute drive from home, which is extremely attractive, versus spending a few potentially weeks away from home. Maybe the difference ought to be more of a shared, maybe the paradigm ought to be more of a shared care paradigm, whereby the treatment is given and the acute toxicities are given, are looked after in the major academic center, and the follow-up, et cetera, it can be given locally. We at least should consider that, I think.
Yeah, that's where we've spent some of our time at the foundation, is digging into long-term follow-up. We ran a patient survey across clinical, commercial, just trying to look at these challenges to see, and what is the way that we can deliver in a patient-friendly way that follow-up. I think to Sarah's point, not every patient is alike. Not every patient wants to drive into the city ever, something I learned over the last 3 years being more patient-focused. While I take my children to New York City at 1 year old and put them in the chaos of this world, not everybody wants to do that. So I think it comes down to the patient themselves, but it also comes down to asking them the right questions around what are some of the hangups, do you plan to follow up, what are some ways that we can make this easier? So an interesting data point from our last survey was that 81% of those 100 patients were comfortable sharing their health records directly with their treating sponsor, their CAR T commercial company, which it's great to see that type of commitment and connection, and we do see that in the CAR T world. There is a different connection to the pharma company making your drug, to the people manufacturing your drug, and to the academic centers and the doctors creating the therapies. So I think there's a lot of work to be done there, and more questions to be asked of our patients. What is best for them, and how do we deliver to that 80%, which they're not all alike, and that's where we'll dig a bit further at the foundation. Not all community hospitals are alike.
Right.
There might even need to be a community that just needs to see Emily's story because they've never heard it. All of us in this room have heard it so often and are moved by it, but there are so many others that haven't. So where do we meet the patients where they are and kind of grow all tides in the community?
Yeah. And I think Sarah's point is really important. I think no one's suggesting that in every single community practice we're going to set up a CAR T, establishment. But what we do believe is there are community mega practices that are highly sophisticated and give very advanced therapies in the community, and that could be very much working in partnership with academic centers and improving coordination of care. For example, one of the big areas we probably should address is, speaking of regulatory pathways, we need to reduce the labels in the current CAR T world, where you're requiring 4 weeks at an academic center and 8 weeks of not driving and needing a caregiver. That is a barrier. We see that most of the fallout, why only 2 in 10 patients are getting access to CAR T when we've done market research and talked to patients and their caregivers, it's that. I'm super excited about the promise of this one-time infusion. I can imagine myself living at University of Pennsylvania, MD Anderson, whatever, but at 4 weeks is tough, and I have to not work. My loved one potentially doesn't have to work. So I do think following the data is super important. And when we know that 99% of all tox happen in the first 2 weeks of an autologous CAR T, why isn't the restriction 2 weeks with a very tight coordination of care back to the community on watching the next 6 and 8 weeks. For me, I feel like we should now follow the data and evolve the class labeling that was put in place at a time when it wasn't as well understood, but with reams of real-world data, I think we now can evolve CAR T and make it more patient-friendly to access this care and not have some of the burdensome label requirements.
Yeah, and I think you all are bringing up such great points about access because, Linnell, to your point, who can take 4 weeks off and move to a medical facility? It's only a certain percentage of the population, and we heard about that a little bit this morning. So we've touched on some of the logistical bottlenecks already. What are, and maybe Sarah, I'll start with you, scientifically, from a policy standpoint, what is in the way right now that we need to kind of clear out in order to get these products to patients?
All right, well, so I guess I will start. I think when it comes to the science of ex vivo CAR T and TCR therapies, people in this room and others have done so much work for new therapeutics, new targets, new patient populations, and we had a really nice discussion about that earlier this morning with respect to increasing access. And the science of it, I think, really relates, at least from my perspective, relates to questioning some dogma, which really is, I guess, another way of saying deep scientific insights into what it is that we're doing. I do see a lot of, there's still a lot of unanswered questions with respect to what is the best CAR construct, what is the best way to deliver, what is the right dose, what is the right lymphodepletion, all of those things that in our very justifiable and successful, I'll use the word rush, but I don't mean that in an excessive way, I just mean our enthusiastic and successful progress towards getting regulatory approval and then commercial and then giving to patients, that there are, kind of similar to what Linnell was saying, some overhanging assumptions, both from a logistical and administrative point of view, but also to your question from a scientific point of view that have remained unaddressed. There could be a lot of, I'm going to use the word tinkering, but I don't mean that in a trivial sense, a lot of additional individually fairly incremental, but ultimately in aggregate probably quite consequential improvements to the system that could be done. A small example, do we need lymphodepleting chemotherapy? Yes, I think we do, but how does lymphodepleting chemotherapy, could it be substituted for something else? Some early data from various sources that may be using, let's say, armored CAR T cells, which I'm sure the audience knows, but incorporate activating cytokine in the transgene cassette, maybe do not need lymphodepleting chemotherapy. Why is lymphodepleting chemotherapy bad? Well, it's yet more chemotherapy to patients who have had a lot of it. It is relatively low dose, but nonetheless, this is for people who've had lots of chemotherapy before, has significant, and we, I think, are learning even more side effects than we were originally aware of. So those are some scientific advances that I think need and can happen quite easily, actually, over the next few years. But one of the problems is, to Linnell's point and to earlier points earlier this morning, that's not the label. That's not currently how that's done. And from an academician's point of view, is that the most interesting and intellectually exciting use of my time and my resources and my diminishing grant funding to do that? Maybe not. But those would be, I think, very palatable for patients if you can say, well, you're not getting more chemotherapy, you probably won't have cytopenias, or hopefully won't have cytopenias, and maybe not, the certain incidence of therapy-related cancers that can occur later. So that's with regard to the ex vivo CAR T, and then finishing off maybe my soliloquy with the in vivo CAR T. An in vivo gene therapy basically is, it's very early days. I mean, there's vanishingly little, if any, real clinical data about this as yet, but it's coming very soon, I think. And so we shall see what the pros and cons are and the degree to which the current systems can be readily translated to the in vivo field, or now from my point of view as an academician and my, and others' true enthusiasm for it is what scientific advances need to be put into place in order for that system to be as effective as the ex vivo CAR T.
Yeah. George, I'd love to have you chime in on that question.
Yeah, I think from a scientific perspective, we look at something discussed in the earlier panel around, and to what Sar just mentioned, there is such excitement around the transformative nature that we almost couldn't pause to get all of the data we needed.
Exactly. Mm-hmm.
And having been in the CMC and manufacturing side at the time, I was a little more concerned about that than I am now on the patient side. Is how fast can we get it there? How do we know the mechanism? And then do we have the funds to go back and retroactively look at that? So I think some of it comes to CMC and product characterization. I guess the unsexy part of research and that engineering side, but really the further we can classify that it is, the easier we could insert a new technology, whether it be automation, whether it be a new vector, whether it be a new platform. If we are clear what that mechanism is, we could sub in, and to Kristen's point earlier, shorten the development time, shorten the cost of development, and perhaps have that path to profitability. But that work has been really hard as innovation, and the outcomes have been so great. It's an unfortunate consequence of such unbelievable clinical data is that you really don't have time to understand as much, and investment flowed so early 10 years ago.
Right.
So how do we go back in time and look at that product characterization point? And there's so many great CDMOs and great engineers thinking about this, but not enough time, funds to do so. So how do we tackle that?
Yeah. Linnell, you've been nodding along this whole time, so I'd love to hear what you think.
I think it's right. It's the tension in the rope. And Kristen Hagee said it earlier, you know fairly quickly when you have a breakthrough, right? So you put it in 10 patients, you sit there and say, we're experiencing this now, and you're trying to race to the next gate. And it's important because you have to balance, and you want to make sure the lesson we learned from our first generation autologous CAR Ts that we have in the market today, we will never not have the robustness and methods baked going into pivotal. Because if you are in flight in pivotal, the idea you're going to change major parts of your process or your methods, you're going to not, and you're going to then commercialize, and then you're going to be living with suboptimal, robustness, which can impact your manufacturing success rates. It can drive up significant expense as a result because you're now not getting reimbursed for the patients that are, out of spec. And so I really do think you have to be very thoughtful on balancing speed and rigor in order to ensure in the end you're thinking about how do I help the most patients at that endpoint versus I got there really fast and I helped a subset of patients, but I could have helped a whole lot more had I take 6 more months and got my robustness, my characterization right, my data package right, so that I could then move from there, because it does impact you for the life of your product, and that certainly can be a long time depending. So I think that's super important. The other thing that's been said here, and I think it's really, really important, is, in a larger company, we can probably absorb different things, right? I do think what worries me most is this ecosystem we're in cell and gene therapy relies on biotechs. And we can't answer every scientific question, and academic center can't answer every scientific question. So the biotech ecosystem has to thrive. And I do worry that if we don't create frameworks for them to operate and interrogate early proof of concepts and advance concepts alongside of academic centers and alongside of pharmas, I do worry, because investors are starting to get a little bit more concerned about how big can CAR T really be. We, after all, have now been in market since 2017, and the class share is only 20%. That doesn't seem like a very viable marketplace for CAR Ts. And so I do think it's really, really important that we remove some of these barriers and create frameworks for biotechs. Otherwise, it is going to be an industry that's going to get narrower and narrower and more consolidated, and I think that's extremely dangerous to interrogate broad sets of questions across a broad set of populations.
No, those are all really good points, and I wonder, as a follow-up to that, we were so happy to have Nicole here today. It was so great to hear from her and her perspective. And what I heard was, which FDA has been saying for a long time, right? Come early, come early. But then I also really appreciated, and this has happened a little bit on the small molecule side back in John Jenkins' day as the director of the Office of New Drugs. There should be not different standards, but different opportunities for, depending on the size of the company, the experience, the sophistication. And so I was happy to hear that as well. But I wonder if Linnell, we could stay with you and then move down to your other panelists about regulatory What do we need from the regulators in order to overcome some of the bottlenecks? We've talked about a lot of that already this morning, so would love to hear your thoughts on what has been said and if you have some other ideas.
Yeah, two comments really quick. I wouldn't create different bars for different size companies, because that will create problems. You want to make sure at the end you have a standardized set of criteria that I just don't think smaller companies have access to information. So I have the luxury of, we have 2 products in the market and we have 3 currently in clinic, which means we have a reams of data and understanding of autologous CAR T, from vector to process, characterization, et cetera. And we have ongoing conversations with the FDA through different IND check-ins, discussions with our commercial products if we hit any challenges on spec widening, vector changes, manufacturing site changes. And so we were informed as a result of the cutting edge thinking at the FDA, because we are in constant conversations, because we have a breadth of assets and a breadth of conversations that need to happen. I do think more democratizing of information is helpful for the broader ecosystem. With protecting IP, because I'm not going to sit up here and say that my shareholders would freak out if I was going to. So clearly protecting IP is critical. But the second thing I would say here that I think is important, but there are some areas of bottleneck. Two of them are raised today. 15-year follow-up on a CAR T, it is cost prohibitive for any size company. This is becoming an enormous discussion point for us. When we choose, do we go put money towards another CAR T, or do I go put it towards a monoclonal antibody, or go put it into a small molecule? I will tell you, this is a big deal. I do think this needs to be addressed because it is very cost prohibitive no matter the size of the company. The second thing is on the specifications. It was brought up earlier, and I think it's super important. And let me first say out loud, the FDA has been extraordinarily flexible and responsive. So I felt kind of bad when they may be given they're not as flexible as they need to be. They've been highly flexible, but they do have to have data, and we have to provide them good characterization. But I will say on specs, when you launch with, or you get approval, you look at clinical data across a broad set of patients with a large tail of spec release criteria, and then we're launching with very tight specifications. And so I do think we have to revisit, how do we actually get the patients that are on the tail, that when we look at data, they have the same clinical outcomes, same safety profile as the people that were inside the specifications. That makes it very difficult for any size company when you're dealing with very narrow specifications, which means you'll have a lot of manufacturing challenges, which drives up cost overall for the whole system.
Yeah. We are coming right up on time already. I don't know how that happened. So George, I'm hoping I can start with you on this last question. If there was one change or one advancement that would significantly accelerate progress in this space, what would it be?
That's really difficult to think of one. On the scientific side, there's obvious freeing up of that bottleneck in vivo and allogeneic and available. But we do have to look at, there are commercially approved treatments, not just for CAR T, but in sickle cell that are cell-based ex vivo. So I continue to point back to the regulatory side, to the last question, to where Linnell's speaking. We've pulled together, after that patient data, a consortium with some partners around some guidance for long-term follow-up. We mentioned the acute toxicity happening within 2 weeks, but within 2 years, most of those secondary malignancies and others are happening. So potentially bringing that together and reducing the cost of development, I think is a key part. And I think that's really universal, whether it's Dr. June wanting to start another trial instead of following up with 20 patients for another 15 years or another 10 years, or Pharma and Linnell's team wanting to start something new in their pipeline versus follow-up that patients don't necessarily want to continue to do. We saw it in the early CAR T trials. The patients that we talked to from phase 1, they love going back to Penn. That's not the case as we've commercially evolved it, and that's some of the data we've shown in that. So really trying to put together some guidance in the form of a white paper in the next couple of months to drive some of that forward, both for cell and gene therapies.
That's great. Sara, do you want to tackle the impossible question of the one development that could change?
It's not that hard, I think. We just have to go a lot faster. Each and every step of the way has to be just a little bit faster than it currently is, so that in toto, the whole process will be much faster, because the only experiment that matters is the clinical trial, a well-conducted clinical trial in human beings, not the animal work. And so it has to be done responsibly, but it has to be started, executed, and concluded, and the data shared publicly as quickly as possible so we can learn from it, because the whole process is way too slow.
Yeah.
Linnell, last word.
I actually agree with Sar, and I would say we should look very closely at our friends in China. They have created a faster path, a proof of concept than this country. They will pass us in cell therapy. I will tell you there are more clinical trials being interrogated in China than there are in the United States. And so we really need to ask ourselves, are there regulatory paths in other countries that could work here on that early proof of concept? Because we're trying to understand really quickly, should we invest in here? Is this the right target? Is it the right patient population? Is it the right platform? I think those early frameworks can be really, really helpful to then say, because investments will follow. You know what I mean? But you got to create the early, fast frameworks to be able to interrogate science quickly, and then allow from there the investments to follow, and follow then the rigorous process we should to get drugs approved.
That's great. All right, well, wow, we have packed a lot into our half an hour together, so please join me in thanking our panelists. That was a great discussion. Thank you all.
Exit stage left?
Jeff
No,
is
we have
on
to
his
go
way
right.
to
There's no exit that
queue up
way.
the next panel.
Thank you.
Thank you.
Thank you, Kate, George, Sar, Linnell. Thank you very much for joining us and for a great discussion this afternoon. We're now going to transfer to our final panel of the day, which will explore proposals to reduce production costs, foster sustainable partnerships between nonprofits, academia, and industry, and support scalable, adaptable manufacturing models. To start us off and provide an overview of these types of strategies to access cell-based therapies, we have Dr. Natasha Kekri from the Ottawa Hospital Research Institute, who is prepared to share some thoughts in an initial presentation here. So thank you, Natasha.
Thank you. Well, thanks for having this Canadian here to join in the fun. So I'm Natasha. I'm a hematologist in Canada, and I was asked to share our experience with what we're doing. We're sort of pivoting now to talking more about the manufacturing, and that is something we've been trying to grow in Canada. So I just want to remind everyone, we all know this, but just to remember that we're talking about different models, all with sort of different goals, but really there's different ways to decentralize manufacturing. Decentralized essentially just means we don't have one main site manufacturing, there can be many. How that is structured in terms of whether it's regional, whether it's really point of care, meaning that it's actually close to the patients, whether that's close to their clinic or their hospital, or a distributed model where you can still have quality oversight in one place, but distributed manufacturing. There are different models that follow this, and so they're not all created the same. I was a hematologist. I trained at Boston at Dana-Farber for my fellowship in cell therapies. I came back to Canada about a decade ago, and I knew these therapies were going to be promising for our patients, but I also knew my own patients were not likely to have quick access to these therapies. And so with like-minded physicians and scientists in Canada, we built a program in 2017 called CLIC, Canadian-Led Immunotherapies in Cancer. And really our vision was to develop a Canadian academic and a manufacturing model that really would allow for the delivery of research and expertise in this space. We thought that that could build infrastructure, it could create jobs and foster innovation in Canada in this cell therapy space that we knew was super exciting. The strategy at that time was to centralize our plasmid and lentivirus manufacturing, but then to create a point of care compatible program for the CAR T manufacturing. And we started with CD19 truthfully because there was already a benchmark of what that should look like, and we knew that that was something we wanted to at least meet in the beginning. Oh, so this got all changed. I probably should have looked at my slides, Mark, but basically the thing that we built back in 2017 was essentially a model where we had manufacturing of the plasmid all the way on the west side of Canada in Vancouver, BC. We had the virus manufacturing all the way on the east side in Ottawa. And then we had one manufacturing site, you'll see in yellow here, in Victoria, British Columbia, with 2 clinical sites on either side of the country. So this is a huge country, as you know. Our population's not huge, but this is a huge geography. But at this point, we started with 1 T-cell manufacturing facility with 2 clinical sites at opposite ends. And we built, using a Miltenni platform for our manufacturing. This was a Prodigy machine that was a closed benchtop system that would be feasible for us and easy to build into our academic programs. And that's why we started with this. It was well established. And our main funder at the time was a not-for-profit, BioCanRx, which is our Canadian immunotherapy network. And that's really how we got off the ground at the very beginning, with academic institutions who believed in this vision and a not-for-profit who believed in what we were trying to build. I'm not showing this data to review the trial or to tell you anything about it, other than to say that we did do what we said we would do. We treated almost 100 patients. We've had great success in efficacy, as well as low toxicity rates. And so we've been pretty proud of the fact that we were able to build this initially. But at that point, we were still not point of care or distributed in any way for our manufacturing. Like I said, we had T-cell manufacturing at one site. And so we had to decide how we wanted to build our decentralized approach. And this is how we did it. We stuck to one quality oversight program that rolled this out to other sites. And we did this by doing a gap assessment of new sites coming on board with us. We then did a formal tech transfer. And then we actually insisted on formal audits from our original site to sort of make sure that we were all on the same page going into the same clinical trial together. And so in terms of decentralized manufacturing, there was, of course, challenges. We were a small academic group with a small amount of grant funding to do this. And so we really had to figure out how will we be able to do this in multiple sites that have their own quality systems. How would we bring that together under one clinical trial? We also have to remember these are different academic institutes, so they have different priorities and different constructs they want to bring into the clinic. How do we make sure that we're thinking about that while maintaining the priorities that we set up as a national program? And of course, funding is short term. We had to think about how are we going to build this in the more long-term way, and we didn't have those answers when we were starting. I don't know that we necessarily have all of them now, but we definitely are thinking about that. So this is what we built as our second version of our program. We now have binder development through the NRC in Canada. Still with our not-for-profit BioCanRx, we're funding this throughout Canada, but we have largely clinical sites all across the country. I didn't include all the pediatric sites that are now onboarding, so that's happening as well. And now we're moving from 1 towards 3 manufacturing sites for T-cells as a starting point. This is certainly not point of care yet, but it's becoming decentralized in our program. And what we have built as we do that is really a pipeline in Canada to allow for Canadian innovation to reach the clinic. And so we now have a GPNMB CAR in for sarcomas that's treated patients that was built through this pipeline, a CD22 single domain llama-derived CAR that's in the clinic, and a mesothelin CAR coming now for gynecologic cancers. And all of that we built using this platform and really through great patient partners and pretty much finding people with the same vision as us, we built this pipeline. And we now have international partners. So I mentioned on the right here, DanCAR-T, which is a Denmark clinical trial that used our vector, our protocols, our system to build a trial under their regulator, really with the same vision that we had, which was to bring access to their patients and bring a system and a pipeline for their own country. And that's been a great partnership for us. So we were able to bring this to the clinic and bring it to clinical trials, but really, as we've been talking about today, how do we bring it past the clinical trials? Well, that's the next stage of this, and that's what I'm working on now, which is market authorization. So really, when we talk about market authorization, we're really figuring out how are we going to do this in the long term, and with who will make that happen. So I think it's tricky, and I don't think I need to again say all of this. We've heard most of this today. But I think we are left to try to work with conventional commercialization strategies from an unconventional platform. So we're working with an unconventional manufacturing platform, mostly with academic groups, and in my setting, a not-for-profit and academic groups who are not well funded to do this, mostly in smaller indications. So again, we're still not there with how we're going to fit that into the existing frameworks. And I don't have to focus on the regulatory flexibilities. We know they're there, but we don't know what they are as academics, and that's part of the tricky part for us in trying to build this into a program that can be sustainable outside of trials. So for us, we have the clinical data now. I think that's great for the CLIC program. But now we have to move beyond clinical trial manufacturing and think about what GMP looks like in the commercialized stage. And that's where it gets trickier for us in terms of figuring out, can we license our facilities that are currently academic and run trials? Can they be licensed to do this commercially? We have to talk a lot more to our regulatory body, which is Health Canada. We have to figure out under what organization will we do this. So right now we work with a not-for-profit, but should we work with a benefit corporation or a B corporation? Should we build a private-public partnership? those are lots of questions we haven't completely answered, but that's what we're looking into in terms of how we can sustain this model. And of course, for us, our funder is government. We only have that one funding option. And so we have to talk to our funder, which is the government, about how we're going to do this in the long term, because I can't put in all this work and ask people to fund this if ultimately we can't get the funder to pay for it. So it's important to bring them to the table for us right now. So I think we still have lots of challenges ahead, but I do think these models that we're going to talk about in the panel can allow for these valleys to be crossed. I think we do still have, especially in the space of cell and gene therapies, it's difficult to get from the mouse models to the clinic and then from the clinic to the healthcare system. And I think you heard Sar say that we really need to make all of that faster if we're going to make this reach patients in a better way. And I think that through these decentralized models, hopefully we can speed up some of those things, but we have to be thinking about innovation in our healthcare adoption systems and in our product delivery systems if we want this to really roll out in the long term for patients. And so I can't stand up here and not say that this is a huge effort. The reason this works in Canada is I have academics who believe in the same mission that I do, and I have scientists and funders who are willing to help us with making this vision happen. And so all of these people really are just a small part of it. And of course the patients, we've talked about patients a lot today, but they do put their faith in us. And it is very hard for these patients because they often have no other options. So they have to put faith that we, on the side of the science and the regulation and the policymakers, are all thinking about them at the end of the day and protecting them, because ultimately they might not have other options. So that is our job too. And I, of course, have to thank them for everything that they contribute to this field as well. So thank you. And with that, I think John's going to introduce our panel, and everyone's going to come up here.
Thank you all. Please have a seat. Well, and thank you, Natasha, for that wonderful introduction. So one thing we heard is Natasha's introduction in the decentralized model that she's created there out of Ottawa. I think it's really part of the focus that we're talking about today is increasing access and using decentralized models to do that, and delivery and working on costs to actually deliver for that. I'm wondering if, as I introduce the panel, you heard about Natasha's operation, perhaps we can go through and Ben, we can start talking a little bit about what you're working on and your introduction as well. So Ben Banansky from Allogene, and please.
Yep. Hi, everyone. Like you said, Ben Banansky with Allogene. We're producing allogeneic CAR T cell therapies for oncology and autoimmune indications. Back to what model, we're currently using a centralized model, but certainly, I think whatever the models are, many of the things we're talking about today are relevant to any manufacturing model when it comes to trying to move efficiently while also maintaining enough consistency and control of the product so that we can make a safe and effective product for patients.
Absolutely. And Boro?
Yeah, Boro Dropulic, Caring Cross, we're a non-profit, and our goal is trying to take autologous therapies, much like what Natasha discussed about, and making it affordable and accessible. And we are developing a network of hospital sites in the US, but we also work with partners around the world. So for example, we're working with the Ministry of Health in Brazil to bring local point-of-care manufacturing to that country by technology transfer, teaching how to produce the vectors, the cells, and so they can produce it locally and leverage their skilled but less costly workforce and labor to really reduce the cost.
Fantastic. Stephen?
Hi, I'm Steve Feldman from Stanford. I think we're a hybrid system. I would say we're a centralized point of care, right? So we're not in the hospital, but we're adjacent to the hospital. But we have to manufacture a variety of programs, and I think that everything that Natasha described, like we have to do as a single institution. So there's challenges there around cost and accessibility. So I'll leave it there, but I have more to say.
Fantastic. And Natasha, we've heard from you, and please.
I'm Kim Schultz. I'm the division director in the Office of Gene Therapies, and I'm trying to figure out how to make all these manufacturing strategies fit with the regulatory framework.
Fantastic. That's great. So, I mean, this is really, Kim, you bring up the key question here, right? So, why is it that we need to think about new manufacturing models? Bora, what do you think?
Well, I think because there is a gap between the technology and these life-saving therapies and the need, right? There are only 25% of patients eligible for CAR-T are receiving it. And I think that where companies are not filling the gap, that's where a hospital-based network can really, I think, fill the gap. So for example, in certain indications where it's not profitable, say for pediatric cancers. But I would extend that to indications that are more general, where, for example, patients with advanced cancer don't get the therapy in time, right? And the hospital can make it within 1 day, for instance, locally. Or where geographically or demographically disadvantaged patients as well. So I think we need this safety net from an ethical point of view. And I think that the challenges that we need to address is how to create the network. This is autologous therapy. I don't necessarily think that we just think about jumping to in vivo is necessarily the solution. And the reason for that is that it all depends upon the dose. We already have direct injectables, AAV vectors, and they cost, in some cases, multiple millions of dollars. So we really have to think about, maybe what works and simplifying it and improving it.
Right. And Natasha, what led you to look at that distributed model that you just talked about, and what were the needs you saw?
Yeah, I mean, I think we can't forget about where we came from, right? I mean, without pharma, we wouldn't be where we are in this field now. But I think what we risk moving forward is that we are worried that the next set of products that come on the market will already be outdated by the time they come to market, right? And so I think these manufacturing models can help us in figuring out certain things maybe faster in a more nimble way than the traditional method that got us here. I think now we are in the luxury of knowing enough about the field to change things and be more nimble. So I think these manufacturing models allow us the opportunity to do that.
It makes a lot of sense. And each of you is in a different kind of sphere, whether it's industrial sphere band or in academia or in government. Could you maybe dive into the models, Ben, that you're working on at Allogene that you have now, and what are the challenges that you're facing?
Yeah, so like I said, we're doing allogeneic, which in some ways is different, right? We can make a product that's off the shelf, hold inventory. Cryogenic supply chains are a lot better so that the delivery of material is very fast. But the challenges there are just the cost of establishing it, which is probably true whether you're talking about a distributed model or a centralized model, of the capital cost to build facilities, the costs to produce, even if you're not doing it yourself, but to do it through external partners, starting materials, vectors, CRISPR reagents. The quality control and testing costs, with these products, unlike traditional pharmaceuticals or biologics, the quality control tests are much more complicated. And for example, our manufacturing plant, the square footage of our QC labs is the same as our manufacturing facility, just to show you the complexity. Sure. And that all has to be built, a year or more before your studies can even start. So there's a lot of upfront investment required to do that, and a lot of complexity to manage, regardless of whether you're in a centralized or decentralized model.
Sure. And Steve, how does that compare to what you guys have at Stanford and the direction that you're going?
I mean, we face the same challenges, but I keep coming back to thinking about the earlier panels, and there has to be ways that we can leverage existing information and data, right? So clearly defining what a platform designation is, disseminating that information to everybody so that we can understand it, and then can we leverage that such that we could reduce time and cost? Like, we have to make a GMP reagent, but after we've used it and we've shown in our phase 1 study that we have positive safety data, maybe even a positive signal in terms of efficacy, when we move forward, like the challenge is like, how do you move things forward? And if we can leverage that existing data, then maybe we don't need to do the same type of PPQ because we believe the existing data means something. So, I mean, I think our challenges are different, right? It's about being able to, cost is always the problem, and how you afford the reagent development just up front of the phase 1 trial is a challenge, in the investigator setting, I think. But then it's that latter stage.
Is that, Natasha, that you're seeing a similar alignment with Steve in terms of not just cost, but being able to leverage the data that's out there?
Yeah, I mean, I would love to go and ask some of these companies for some information, because we're all struggling with some of the same things. Well, let's talk about manufacturing failures, which we don't talk enough about, right? And there is probably a very large amount of information to predict who's going to fail manufacturing. And I don't think that's in the public domain enough for the next set of developers to use that information moving forward. So I think now we're at a stage where we should start asking those questions to people who may have that information. And STAR talks about lots of examples of things that we know in the scientific space isn't maybe as exciting, but there's probably already some data on that we could now use in our systems to grow more quickly.
Absolutely. And Kim, just jumping to that same question, a lot of these problems are, I think, there may be misconceptions or challenges from a regulatory standpoint. What are you seeing as those sort of key misconceptions or challenges that are slowing down the cell therapy manufacturing space?
Yeah, that's a good question. We have guidances out there that talk about phase-specific implementation, for instance, of cGMPs, but we obviously can't touch on every little nuance that would be there in those guidances. So I think talking amongst each other is a great way to share that information to work through those problems. But I'm going to give the normal FDA answer and say that we're always available for meetings, and that's where you can ask those specific questions in the context of your product development and your actual facility capabilities. I mean, to Ben's point, we have Type C facility meetings so that when you're planning your facility, you can come talk to us to make sure that there's not something that sticks out to us before you actually go in and finish building it and putting that together, to kind of de-risk that part of the process. And then even coming in at early phase studies, if you have cGMP questions, you can put those into your pre-INDs. If you need information on room classifications or that type of information, that's all fair game for part of those pre-INDs to make sure that you can get that IND started on a successful foot.
Steve, any thoughts?
Oh, yeah, sorry, you saw my face. I mean, I think it's great, and to be able to interact with the FDA and ask these questions is critical, right? But I guess my question is, how can we create a mechanism to disseminate that information in a de-identified but broader space? Because it appears that it's a sort of a case-by-case basis, right? So I think FDA is flexible, and I want more flexibility. Like, how do we disseminate that we could leverage?
So how do you know what the baseline is of that flexibility? So as we said earlier, we can't go out and tell everybody else's trade secrets. That's not where we live. But we do have mechanisms where we try to answer questions in a large format. So for instance, our OTP town halls, those are places where people can ask very pointed questions, and we can give very specific information that can help perhaps interpret some of those guidances, so that guidances that were made for all products, so that you can hear our interpretation of it for cell and gene therapies.
Makes sense. Bora, just a quick question on when you look at decentralized manufacturing, what are those biggest cost drivers that are out there, whether it be infrastructure, training, you name it, in that setting?
I think it's labor is one of the big ones, right, in terms of the QC testing, and when you couple that with the manufacturing. Sure. But also the one single biggest material cost is the vector itself, right? So centralizing that, right, but then I'm thinking about how to make processes very simple or more go towards more automation. That's the way to really reduce the overall cost.
Right. And Steve, that's also reflected in like an academic-led model as well, the same kind of cost drivers when we think about it.
Yeah, labor definitely is a big cost, but again, it goes back to these platforms, right? If we're using an established technology, is that a platform? If we're using, and even on the vector side, right? If we're using a vector manufactured by the CDMO, sure. If we're using their backbone, I mean, if it got platform designation, would that lift some of the burden on us to, downstream of what we need to do in terms of cost or comparability? But I mean, yeah, it's the same drivers. I mean, and then as you move down this process, right, it just goes up. I mean, more quality assurance, more quality control, more people to manufacture. So
when you're implementing this, Steve, in the rare disease setting, are there any unique manufacturing challenges there, or is this the same?
I mean, it's the same, except that, I mean, it's been brought up by many, earlier in this meeting, is that it's the cost, right? At that point, right, who pays, right? Or how do you pay for, even a pivotal trial? Like, could that be done in an academic setting? I think the answer is yes, right? But does it require more? It's going to probably require more on the quality side, and but I think, like the FDA, it was, has evolved over time. I think academic manufacturing facilities have also evolved. So where we were 10 years ago is not where we are today, and we can develop and provide a data package, that's fairly robust with the, CQAs, well-defined, things like that. So it's, but it's that next phase, like how do you move to the pivotal trial and/or commercial, and can we do it? And then, yes, could we do it commercial? I mean, I think there are case studies out there where people have tried, the burden is onerous, but then it's about how do you do commercial and non-commercial in the same place. But
yeah, one way to think about how to economize on a decentralized or point of care model in terms of cost is to think about it more as a hub and spoke model, where you have a reference facility that does all the centralized training, holds everything. Even some of the quality functions could be centralized, with more of the other facilities are sort of more manufacturing and testing. So that's the model actually we're approaching it in Brazil, where you'll have a centralized facility and more the other hospitals will actually just simply manufacturing and test, and through the web, you have a more of a centralized way of approving products real time.
And you can think of this as, Ben, please go.
I was just going to say, adding to this discussion on cost, I think one of the biggest drivers, once you have a facility, is capacity utilization at that facility. And that's one of the big challenges. If you had a facility that just made a couple doses per year for a rare disease, those doses are going to be very, very expensive. But whether it's centralized or decentralized, if you can sort of accumulate the demand and have a fully utilized facility, the difference in per batch or per dose cost could be an order of magnitude difference. And so that's where some of the discussions about partnerships or academic centers that are maybe producing many different products, the more we could think of models that maximize the number, maximize the capacity utilization for these very expensive facilities that we build and operate is going to be a bigger driver than raw materials or labor or anything else.
Absolutely. Yeah, I like that word partnership. And Natasha, what are the partnership models that are out there that could work in this setting, whether it's cooperative hubs or private-public partnerships?
Yeah, I think what we know is there's so many components here, right? We're talking about plasmid, virus, cells, and then we're talking about quality systems. Yeah. And at each of those components, you can have partnerships, whether they're public-private, whether they're multiple academic collaborations, even what we did with Denmark, for example. And there's companies who are willing to do certain parts of that, right? We've seen some mobile apheresis. We've seen mobile quality systems that come in. I think, much like Boro, we're trying to start with one and build a hub and spoke, but truthfully, that's still incredibly expensive for us. And as Ben said, it has to be a minimum number of products made for that to become cost effective for us. And for us, we have to plan that way. So we are planning with knowing our numbers and how many patients we would need to get into the pipeline for this to make sense from a money point of view. But I think those partnerships are going to be how we build this. And there will be smaller firms and smaller biotech who have interest in these specific components that I think will actually really change the game for a lot of our type of work and the academic work that we're doing.
Yeah, sure. Bora, in a partnership like that, what do you think the key metrics are that are going to ensure success of that kind of partnership?
Yeah, the way we view partnerships is more licensing out and partnershiping with local organizations. Okay. So for example, we have a partnership with the Burjeel Hospital Group in the UAE. Sure. Where basically we're going to be licensing out some of our vectors so they can produce it locally. So that's one type of partnership model. But the other partnership model are networks with hospitals in which you actually, as a common goal, go towards an approval of a product, but then weight the relationship towards the hospital. So the hospital actually has the majority of the benefit. because that way they're incentivized to be participatory and take on the risk of being part of such a network.
That makes sense. Makes sense.
And just to build on something for partnerships, right here, this is a partnership, right? Like something like what the Friends is doing and bringing together stakeholders from regulatory policy and academics, I think that is going to be how we can move some of this forward, because I think even just in my conversations at lunchtime, there's so many things that we can do together as a community. Even with regulators, you've heard multiple times that they're willing to talk to us. So I think we have to, even this kind of a partnership can really change things.
Yeah.
I mean, I think partnerships are important. We explore partnerships all the time. You know, and it's really, there are partnerships around reagent development mostly, right? But I guess, and so they're valuable, right? It comes back to the platform designation stuff, and can we leverage that? But then, you know, what happens next, right, is that, you know, the partnership to license a technology from an academic center or to commercialize something, right? That's the real gap, right? I mean, there's lots of interest to partner around making vector or making plasmid or guide or whatever, but not that next phase. And so we need to figure that out. I mean, these hub and spoke or any of these models, right, that you use, I mean, still, they still require trained staff, you know, a robust quality system, you know, and then like the liability associated with that. Like, how do you ensure that your spokes are trained, you know, and manufacture the same product, and do you need to do the same comparability? But the partnership that's lacking is the next phase, you know. If we make a successful vector with somebody, you know, the idea is that, you know, if it moves towards commercial, the pull-through is that they manufacture the commercial vector, but who manufactures the commercial product, right? So that's the key. We still haven't figured out. I mean, that's particularly in the...
Is there an opportunity, I think, you know, particularly with like the way research funding is right now, even from early stage and in translational research funding, is there an opportunity to begin to create these partnerships very, very early so that you had line of sight, even all the way to commercial, so that you had some buy-in from either the biotech companies or pharma to say, we're going to invest in this project on a given indication, and we've got commitment all the way across, not just to say the phase 1 trial?
I mean, the short answer is yes. I mean, earlier engagement on all things is better, but I don't know, I just struggle to see how that happens. I mean, I think, you know, if we could partner with pharma, if they're going to be the ultimate developers or commercializers of these therapies, you know, moving backward, it's hard, I think, for them to move towards the academic. And then if you think that a lot of, you know, there's a lot of innovation that come out of the academic setting, you know, where we got to do the phase 1 trial and develop all those reagents and then, you know, show some efficacy to create that interest. So I think engagement early is important, but not quite sure how to facilitate it, I guess.
You know, there are some examples out there of cell and gene therapy CDMO organizations that are partnering with academic institutions to operate their manufacturing facilities roughly on campus. And, you know, that's maybe a model that has that early engagement for the, you know, the preclinical and clinical work, but then there's a partner involved who could take it maybe to a different site, or a partner who has more of that commercial experience. So I think there are some examples of different models being tried out.
Please, Barb.
In our partnership with Fiocruz and the Ministry of Health in Brazil, we have a clean line of sight because the ministry is supporting the project. The ministry, in the end, will be the payer, right? So when you have the payer interested and invested in supporting the project and driving it, because in the end they see the benefit, the financial gain, you can have a clear line of sight. So we just need mechanisms like that in this country and in other places.
Sure, and it's supported by, you know, regulatory mechanisms that can see the same line of sight all the way across. And so with regard to that, Kim, so just a question on, like, as it relates to similar to, say, accelerated approval that we see with evidence-based, could there be a risk-based framework that's applied to things like manufacturing as well that could give you guys some assurances and make things a little bit faster?
I mean, I think that what we try to do is to have that risk-based approach early on. I think it would be helpful and to sit down and think about when the changes would be made, right? So as we all know, there's oftentimes that gap when we're moving from those early phases into those late phases. And it doesn't matter if it's an academic sponsor or a commercial sponsor. We know that there's changes going to be made, sometimes a complete change in facility. It often is associated with the manual to more automated manufacturing. And so understanding how that change is going to be rolled out and where that's going to be in relationship to the clinical study, I think would be important.
Yeah, absolutely. And it is a challenge. As you're manufacturing, you're moving, you're doing transfer of technology, for instance, often from academia to industry. There are necessary changes that are happening. So, Bora, what are some of those changes that you've encountered in the past, and what are the applications that we can do early on to try to improve QC/QA frameworks, for instance?
Well, we approach that all the sites that we're working with have to use the identical materials and identical workflow to make sure that we have a consistent products amongst the sites. The biggest challenge will be to link the quality systems between the hospitals, right? That is a heavy lift, but I don't think it's an insurmountable heavy lift. I think there needs to be a willingness with the hospitals and the right funding to be able to make that happen. But that is probably the biggest challenge in linking up the quality systems between the hospitals so they're all talking to one another. And the data sharing, of course, is going to be very key for any kind of a network.
Yeah, Ben, in industry, you're seeing the same thing, that these changes are always a challenge, but how do you address them?
Yeah, I mean, I think one of the key challenges is how do you generate enough data that you have confidence that your change doesn't have an impact to safety or efficacy of the product, but also understand that especially in early stages, the cost of that data generation could be quite high, perhaps so prohibitive, like the example that Crystal shared this morning with, we have to make multiple batches of vector at the cost of millions of dollars each, which could be even, I was sharing this example earlier for an allogeneic therapy where a batch of vector might make 100 or more CAR T batches, and each CAR T batch makes 100 or more doses. You could be then making tens of thousands of doses just to generate data. So I think the opportunity there is also what was discussed on the panel to lean into the platform or advanced manufacturing technology approaches, some of the programs that are already out there that maybe allow you to not have to generate product-specific data, but leverage data you have using the same manufacturing or quality platform.
Yeah, that's absolutely essential.
Yeah, and if I can just speak on that, the designations are great, but we're fine with leveraging data outside of designations. It's just that you have to bring forward, the information on why that specific manufacturing is applicable to yours. So to your case in point, if there's a CDMO who's making a vector for multiple products, you have to work within that partnership of the CDMO and those sponsors to be able to share that information between your groups so that we can have that information in the individual files. But we're interested in leveraging across different products if that information is available.
That's great. I mean, and that kind of flexibility is really essential, even outside the designation. So what is it that you guys would be looking for? It's always a case-by-case basis then? Particularly with comparability, QC,
for the comparability. So, I think that there's a couple different ways to do this. So, we've talked about comparability protocols. That's post-licensure, but we do the same thing pre-licensure with people. Send in your protocol for your comparability study, and let us look at it. I will tell you that there are times that we tell people to take whole parts out. I know that this sounds crazy, but as an example, so for distributed manufacturing, moving from 2 facilities to 3 facilities, we got a comparability protocol that included all 3 of those facilities to show equivalence between sites. That's not what we want. So it's in the CAR T-cell guidance where it talks about multiple manufacturing sites. We really recommend that you assign one of those sites as the reference site, and then you just do comparability back to that. And through there, you'll be able to gain a lot of information about your manufacturing consistency with that. We've seen some people do some really early split runs for their engineering. You have to run engineering runs anyways. Why not try and get as much information out of that to understand how consistent your manufacturing process is outside of that starting material variability? So I think people have a lot of really good ideas about what could be done early on to inform later changes. And as Natasha said, if that information could be shared between the groups instead of everybody having to run the same studies, that's going to make it quicker and cheaper.
Yeah, absolutely.
Yeah, we've already made those mistakes, so we don't need to do it again. And I've lived exactly that, of trying to figure out how you're going to do it and how to make it more streamlined. So I couldn't agree more.
I think this forum really is, we talked a lot about partnerships, a place where we can begin to share that data and get it out there and really coordinate. From a patient perspective, Bora, what is it that, that you're looking for from, from a point of care manufacturing, what is it that those patients are looking for, and what does success look like?
What those patients are looking for is also proximity, right, for care, and obviously products that they feel confident in, right? Yeah. Those are, I think, the major things.
Right, absolutely. And so a lot of this is also time to care too. I think that's another big one too. Yeah. Steve, is that aligned with what you guys are seeing?
I think time to care is always a factor, right? So the shortest process, and can we streamline the whole patient experience, from enrollment through apheresis, through manufacturing and infusion. And I think, we can work to do that in the academic setting. I mean, we just do it, and try to make it as short as possible. I think it probably gets more complicated when the more, the bigger the program gets and if it's moving towards commercial, but yeah, I think it's the same.
Yeah,
they just.
And I'm not sure though, we talk about distributed manufacturing as being the thing that enables faster treatment.
Sure.
But I'm not certain that it necessarily does, or that it's even the biggest driver. I mean, like I mentioned, cryogenic supply chains are getting better. Yeah. We can ship frozen product quite quickly. We're getting to the point where the long pole in the tent is the time it takes to do the manufacturing, or the time it takes to do the QC release testing.
Sure, that's it.
More so than the time it takes to move the cells from one place to another. And so as we think about these models, I think you have to look at the whole sort of ecosystem around it and what might be being close might save a little bit on shipping, but you're adding all of the extra effort and cost to maintain consistency everywhere. So, I guess I would challenge a little bit that distributed is always going to be faster.
Right.
I would just challenge, I respect that, but I would think that if it's inter-institutional, that's always going to be cheaper and more economical than shipping it outside the institution.
Yeah, if you have one organization that's managing everything, there's definitely efficiencies.
Well, even in a hub-spoke model, if you're manufacturing locally, even a hub-spoke model, I still think it's obvious. Yeah.
Yeah, I think recognizing though that the quality issues are still there and always going to be a major challenge. What are the technologies that are out there, Ben, that you see that are on the horizon, that are there to kind of change things and revolutionize things?
I mean, automation has come a long way to where these were sort of very manual, bespoke processes. Sure. In the beginning, and there's more and more automation, robotics, and all sorts of things that are making it more consistent. That hasn't gotten that's covered the cell manufacturing part, but I think there's a huge opportunity in the QC space.
Yeah.
So even if the manufacturing, I saw a picture of the Prodigy, and that doesn't test anything, right? That's just doing the cell manufacturing part. There are cell-based assays that are just as manual. So I think there's a huge opportunity for automation to find its way into QC testing as well.
Natasha, you seeing the same thing?
Yeah, I mean, just with these models, we can do more on the cost innovation, manufacturing innovation, and delivery innovation, right? That's kind of what we have the benefit of doing in these systems, maybe figuring it out and then, changing it for the longer sustainable plan. So I totally agree. I am now with a model that I like. It's feasible. I ship fresh product thousands of kilometers, make this work, 15-day turnaround time for in and out of patients. So it works, but I would love to innovate more on it, and it is tricky once you build a model, even in the academic space, to change it. Yeah. But I think that's what we're seeing, though. Biotech is pushing that forward. They're going to bring in new innovation. The Miltenyi Prodigy is now one of many of these platforms that exist, and so we can look at different ones, and hopefully in the academic space, compare them and see what works and what doesn't work.
Absolutely.
Automation is very useful when you have very complicated processes, right, to automate. But another alternative is to simplify the process.
Sure.
And many of these like one-day processes are extremely simple,
Right.
right? So that's another way to go after it. And in that case, automation may not add much to that simple process. And particularly when you think about if there is a high cost to that automation, that becomes untenable in countries like India. They would rather manufacture with their people a short process.
Yeah, it makes a lot.
But I mean, if you move to a shorter process, and maybe if you're doing it at the bedside versus
Not at the bedside, yeah.
I mean,
Yeah.
but so automation would provide throughput, right? If it's around the clock and volume's an issue, but I think just to the point, we need faster or different, release assays, right, that are amenable to automation that we could then have so rapid release, right? And that needs to be a more consistent inline analytics where we could understand our product, how it's doing, and maybe we can leverage that as part of safety data for release, and then, and just automation in general is going to be key. But I mean, it's just, it brings up this question of the shorter and the bedside.
I don't disagree. Automation in release testing, absolutely. The more the better. Yeah.
But simplification there as well. I think that's a great point. Automating something complex is still complex. Yeah. And if we can come up with simpler processes, or instead of needing, an 8-day cell-based assay, if there was some, flow panel we can run that we knew was predictive of the outcome of that assay, and other advances, that's probably as more valuable than automation.
Yeah. And Steve, you brought up, I think, an excellent point in terms of the challenges in the release criteria and the critical quality attributes of the cell product that are coming out. And Kim, how are the agency seeing the application of machine learning to these kind of inline analytics to try to capture cell quality?
We'd love to see it. I know that sounds a little facetious, but it's true. I mean, I think that we're getting to the point where there should be so much information coming out of your manufacturing. And as we said, how could that be leveraged in order to inform better success rates for manufacturing, more consistent manufactured products to try and reduce the impact of that patient variability on the product that's coming out, and how could that impact the success rate for efficacy even. Yeah. So I think that there's a large amount of data that we're just on the cusp to trying to understand, and it would be very useful. Once again, I think that's something that the field is going to have to share.
Yeah.
And so, that's going to be a wealth of knowledge that could help raise everybody, and how that's going to be uniformly put in place, or if it could be shared publicly, I think that would make a big difference.
Fantastic. We talked a little bit in the last session just about disruptive technologies, whether it's your allogeneic cell-based approach, but I think one of the big ones that you heard Sar talk about was in vivo CAR T. And so, is that something that you guys see? What are the challenges of implementing that moving forward from an industrial standpoint or even academic standpoint? Is this something that you guys are actively pursuing and pushing forward? And how do you see that changing a lot of the work that we're talking about today?
Maybe I can take it first, yeah. I think we need to go down parallel paths, right? I mean, I think with in vivo, there's still a considerable amount of risk of whether it's feasible or not. Absolutely. I mean, the issue of conditioning, right? I mean, it's very difficult to consider you'd have to have an amplification system for the cells in vivo, either cytokine-based or another based, in order to amplify those cells. So I would recommend, with any technology you have risk, but you should also go in parallel paths with what we know works, right? And drive that forward and decrease the cost in making these things available. So, the immunogenicity issue is also an important thing to think about.
Yeah, I think it's less about the manufacturing side of in vivo CAR T is kind of there. It's more of the biology side that we're still trying to understand. And until we get to that point, all of these types of therapies are still in play and have that, they're saving patients' lives now. So how do we, keep these moving so that as we figure out more about the biology and what different approaches, whether it's allogeneic or in vivo, come along, because they all leverage many of the same types of manufacturing platforms. So as we make all of those more and more efficient and come up with more efficient, cost-effective manufacturing models, those will be applicable. We're doing allogeneic CAR T cell therapy, but our cell manufacturing steps aren't that different from autologous, and our vector manufacturing isn't that different from what you might do for in vivo. So there's a lot of commonality across all those platforms.
I think something we have to think about too is that we often talk about CAR T cells as CAR T cells, but right, they have different targets. They're for different disease indications. And so I don't think it would be surprising if in vivo works for something, ex vivo works for something else, allogeneic works for something else. It's just figuring out where those different pieces of the puzzle comes together in order to be able to provide access for patients and therapies that work.
And I don't think we should necessarily have two boxes, in vivo versus ex vivo. I think there's a tremendous opportunity right in that space. Short ex vivo that maybe is more like in vivo in the sense that you're adding vector and immediately infusing. I think there are opportunities there as well. So we have to see how the technology evolves.
Okay, sure. And so we talked a lot about both distributed manufacturing, some of the challenges that are out there, improvements in release criteria, quality control. Or just, again, getting back to that concept of a patient-centric kind of view, how many more patients would we be able to serve if we implemented all of these different changes that we've discussed today?
I think that through the local hospitals, you'd have much more access. Patients would get treated. There'll be an increased access towards patients because it's a local ecosystem. Sure. Patients could come to those hospitals more readily. The fast turnaround times and the reduction in cost all add up, I think, to an equation that I think it's a necessary network to create in order to make sure that those products that are not available for patients can become available for patients.
Yeah. And just a question for,
I mean, like how many more patients can you treat? It's a big question, right? Because it comes down to this things we've already talked about, which would be capacity of the spokes or whatever, right? I mean, if you have one automated platform or you need two, like how do you, I mean, there's gonna be rate limiting steps, which are really how many products can you manufacture in a given time. Absolutely. So it may increase accessibility, but it may or may not increase the number of patients that get treated, I think.
That's a good point.
Right, I mean, you got to scale.
Absolutely. One quick question, just sort of round things up a little bit. If you guys could pick, say, one tangible next step that you think is the first thing that needs to change, Ben, where would that be? I guess it's a question for everyone.
Yeah, I guess I'm not going to pick something new because it was discussed in the first panel and even a little bit in this one, but the extent to which we could leverage all the regulatory flexibility that's there and talk more as an industry about examples of that so that, my approaches can be informed by approaches that others have taken. I think people aren't always willing to share where they've gotten flexibility because maybe it's considered too secret, but in some cases that could help us all be more flexible. So some way of democratizing, I think was a word maybe that was used earlier, that information would be really helpful.
Or what do you think?
Yeah, I totally agree with that. I think that's the most single thing I think that'll be most useful to push the field forward.
Absolutely. Steve?
I mean, I agree with everything that's said. I have a question, like on the regulatory side, though. It's as the receiver of all the data and information, and I think I recall when like the first 19 cars were coming out, the idea was that all the manufacturing data and patient data would have to be submitted to the FDA, for follow-up and other things. But FDA is sitting on a huge data set. And so, I don't know, just somehow could we leverage that? I mean, and take out everybody's IP, but there's just a ton of information that we could all benefit from. I mean, I don't have an answer, but I just...
And we did try to do that early on. So that was actually my commissioner's fellowship program. And there was a lot of issues with the manufacturing data being able to put the information together. So we don't have information on the comparability of the different assays between different manufacturers. And so it's difficult to be able to synthesize all of that information into one. There's different eligibility criteria. It wasn't as uniform as we hopefully could have been. We did look across things. There is an article that has been published on it, but I think it was a lot more difficult to do that meta-analysis than everybody had hoped to in the beginning. And also we were restricted to safety. And so we could not look into efficacy.
Could this be an opportunity, though, particularly with the applications we talked about with machine learning, for you guys to actually be the ones training the models to look for early safety signals or even efficacy signals, given the fact that you've got access to all the data?
I mean, I think it would be an interesting question. I'm not sure if we're able to do that, though.
I understand.
Well, and to your point earlier that you have access to information across many products that are very different, that treat very different diseases, it could start to get quite complicated to apply learnings from one to another, maybe.
Could be, yeah.
I mean, I think we've talked about this all day and pushing for that from the FDA, but I think in this room, there's so many other stakeholders we could ask that question to. And it might be time to start pushing our community to start sharing more of that information.
Right on.
So I think we are a community of people who work in academics and also work in biotech and also work in big pharma. And all of those players can come to the table and say more than they do now. And I think it's on us to try to figure that out. And I think Crystal kind of said that this morning when she said, I'll take that back to our community. That is our community. It's still small enough to think about that together. And there's definitely going to be some people in places where you don't expect, even in pharma, who I think will want to do that so that we can make this field move faster, because that still works in everyone's favor. I think when I started this, we kept saying it was like them and us. That's not really what it is. We all have to exist. This won't work if we don't all exist in this ecosystem. So why can't we learn together and do better so that we are making every single step faster? I think that's what's got to happen.
Yeah, that's fantastic.
I think academic networks can do a lot to catalyze such of that openness so that others would be then willing to participate once there's that critical mass of data that's available.
Yeah, absolutely.
But let me just jump in there, right? I mean, in California, we tried to, through CIRM, create an academic network. I think we're still trying to. And it's really defining a network and what the network does. And then we run into the same problems, though, about sharing and IP, and at least we have so far. So it's a good idea, but in practice, it's been a little harder to implement
You have to have a willingness.
than I expected.
Yeah, you have to have willingness.
Yeah.
You can understand how IP can be, but it depends upon the goal, right? I mean, I think you can work out those IP as long as it's shared correctly. But yeah.
But yeah,
But
so
once lawyers get involved, obviously.
Yeah, but we could look at how we can de-identify things and share data. I mean, we certainly want to do that, and we could do that across a network. So
that's a good start.
Yeah.
Yeah, I mean, I started my career working in protein therapeutics and antibodies, and 20-something years ago, everybody thought they had an antibody process that was unique, that was the special sauce, that was their thing. And now we've got to the point where antibody production is industrialized. Everyone's using just about the same process, and those things we thought were our secret sauce really didn't matter. So that could be the case here too, and maybe there are ways of us to collaborate more to industrialize these processes in general.
Absolutely. There's also roles for, from my seat as a nonprofit, there's roles for Friends and for PICE and for other organizations that are network-based, funding lots of scientists, at least at the academic level, to try to pull that data together. And I know there's efforts at Stanford to do that and other universities. And so pulling that data together, sharing it, and then putting it in the hands of people that can really pull out those key metrics and qualities is really essential. And it is probably too much to put on the FDA. It's kind of in our shop to get it done. So I think we're over time. Okay, so yeah, some questions from the audience. Okay, so from Zena, regulatory strategist, with ideas of platform and democratization, are there thoughts on balancing intellectual property? Yes. Yeah.
I mean, I guess
with data sharing in pre-competitive space. So this may be part of that partnership model, and maybe picking on what I just said in the nonprofit space, which is there a pre-competitive area where we can actually work together before the commercialization mode to begin to share data?
I mean, for us, BioCanRx is our not-for-profit Canadian immunotherapy network that is working with lots of global partners that are not-for-profits. But I think while we can work together and come together, I think there's a big piece of that puzzle missing, which is the ones who have already done it.
Yeah.
So most of these academics that we work with that are partnered and have the shared vision that we have, much like Bora talked about, the same vision,
Right.
I think we're also coming at it from a disadvantaged point of view in that we don't have success in this area, really, in the commercialization space. And so I think we do need other partners and stakeholders in there to help bring this forward. I think it has to be, as I've said many times now, both sides to do it.
In our partnership models, we always have clauses that there is a right for the network to non-exclusively have access to whatever intellectual property is being generated through the network.
Yeah.
So having that open aspect to it.
So there's a question on, are there examples of manufacturing failures that the panel is willing to share? I'm sure there are many, but what are the learnings that have come from those failures?
I mean, we're very transparent in this, so it's easy for me to say I'm not tied to any company. So for me, when we started, we tried to use as much as we could learn from the transplant world, because that's where I come from. I have fresh products all over the world, right, that we bring into our site and we give to patients. And we tried to learn from that, and then we tried to make things work for our program. But what we realized was our shipping, which is thousands of kilometers/miles, to get to our manufacturing site is far, and it gets hot or it gets cold in the plane, and we have temperature regulators. And we didn't have robust enough strategies early on for that temperature regulation. And we in our first handful of patients, we had 2 failures that made us go back to the drawing board. And it was a very simple solution and something we could have sorted out probably earlier, but we hadn't planned for. So there's very tiny little things that, again, if we shared in this community so much more, this would have been fixed before we ever saw it as a problem. But I've had other, I think most of us would agree that T-cell exhaustion is a big player in this manufacturing failure landscape. Again, I think if we had more robust data from those who have had more numbers and failures, we haven't had enough failures to analyze the data in any real way. But I think there's lots of data out there that we need to look at and see if we can do better for our patients, truthfully, because if they don't manufacture, that idea that we can reapheresis and do it again, in my opinion, or my experience with my patients, is not likely to happen.
Absolutely. That's a great example. We, discovered exactly the same thing, so that the temperature and shipping is essential.
I mean, I think there's a couple categories, and there was one that was alluded to during the lunchtime panel. Setting specifications. So failure to meet specifications is a manufacturing failure, and often those specifications are set on a relatively limited data set. And as you get more experience, you have some batches that might go outside of that range. So, how do we think about that failure? Is also how do we think about setting specifications, and that could also be an opportunity to be leveraging data across multiple products to inform product specifications.
Absolutely.
Maybe there are some attributes that aren't specific to a product that you could then use a broader data set to set, and then you'd have a wider spec range and fewer failures. I think another category of failures, especially in CAR T, are where your cells from your donor just fail to expand for some reason. Mm-hmm. Very different for autologous, where you don't get to pick your donor, than for allogeneic, where you do. And so for allogeneic approaches, it then necessitates a process for donor screening and selection, so that you're more likely to have a donation that will result in successful manufacturing.
Absolutely. That's a good point.
Yeah, my manufacturing team hates the word manufacturing failure, because there's failures and then there's out of spec, and they're not necessarily always the same. So it's important to think about that.
Okay. I think we have reached time. So with that, I do want to thank the panel and close this off. So thank you very much. I appreciate it. Fantastic. Oh, we do have a Q&A still left that hasn't come up yet. Oh, no.
There's many.
Stay seated.
It's funny.
No, it's the same one, guys. Oh, no problem. Well, it's okay.
Oh, there you go.
So this is a question on AI. So how are each of the panelists thinking about the use of AI during the manufacturing to reduce costs, for instance, during your manufacturing process? Yeah, I can tell you we've applied this in some of our companies before, and I've seen it actually has exactly to do with failure. Some of our processes are a little bit long, depending on like a TIL process, for instance. It takes longer to manufacture. trying to look for early signs of failure so you can restart the process. This is something that has worked well for us. And that's basically using neural networks to look for early signs during the culture that you're going to have a failure on day 3 and not, week 3.
Yeah, I mean, I agree with you. I think, but we just don't have enough data yet, I feel, to really drive an AI model to look for that, like in the academic setting for a particular CAR indication. But I think it's something that we all are thinking about or trying to work towards. So, but yeah, it just takes more data.
This is what we need.
And so we've been talking about AI for a while, and I was at a manufacturing conference a few weeks ago, and it was the first conference I was at where I heard concrete examples of how it was being applied to manufacturing, and these were boring examples. Like I can take all of my standard operating procedures and put them into a large language model and create an AI tool that an operator could ask, how do I do this? And it will immediately take them to the right reference in the right SOP. Or how do I look at all of my regulatory Q&A to help me understand what's the best way to position a certain type of question or response? So not machine learning or looking at scientific data, but other things that can make us more efficient through much easier use cases of AI. I think we have to focus on the that easy stuff too.
Yeah, sure, it's big. And then from a regulatory standpoint, so how do you see the opportunities for AI in your job at the FDA?
For us to look at data?
Yeah, for data review, or what's the biggest opportunity?
Yeah, so we do have experts at the FDA for AI who work on it. I am not one of them, but they're looking to understand how we can use that data to further the field or to further our processes.
Okay, that's great. Fantastic. Well, with that, I just want to thank everyone once again. It's a great panel. Thank you. Thank you. All right. So we've had just a fantastic day of incredible presentations, and this last panel, I think, highlighted one key point that highlighted the actual applications in the manufacturing space. But one thing that came away from looking at the earlier talks, both the lunch discussion about the future and directions, and then the implementation of the technology we've heard from this panel, this is what really struck me is has really been this idea of a partnership and a community. I think something Natasha just said very well is we are this community that's here. We have the data that we can begin to share, and we can pull this together. And we're also the same people that are pointing in the direction of next generation technologies that will address the challenges that we're facing every day. So, so just to close this off, I want to say that another term that you just heard on the panel, but you'd heard earlier, is this idea of line of sight. Like, what is it that we're trying to accomplish? And who are the key players that we can pass off to, that we can work closely with, from the academics coming up with the next generation therapies to the translational researchers and the clinical trialists, the biotech companies, and the regulators, all the way through, creating that community and having a clear line of sight on what the goals we want to accomplish are. And I think that this really starts by bringing that community together. And so with that, I want to thank our partners and friends, the incredible work that has gone into building this meeting out, the workshops that we've done throughout the year, each of our people in academia, in industry, in regulatory. I just, complete thanks that we can bring all of us together to kind of highlight these key issues. And so with that, I just wanted to close out the session. And from Pisces standpoint, from Friends standpoint, I just want to thank everyone for such an incredible and wonderful meeting. So thank you very much. All right. Great.